Welcome, everybody. My name is Rich Hatfield. I'm a senior conservation biologist for the Xerces Society for Invertebrate Conservation. I'm based in Portland, Oregon. I'm happy to have you all joining us today for a webinar about finding Franklin's bumblebee. I am joined by Leif Richardson, who also works for Xerces, and Jeff Everett, who works for the Fish and Wildlife Service, and Hilary Sardinius, who works for the California Department of Fish and Wildlife. I'm going to let them introduce themselves in a couple of minutes. But first, before I do that, I'm just going to sort of introduce Zoom. If you've never used Zoom before, and some of the features that we'll be using throughout the day today. We're probably mostly familiar with Zoom, since this is where a lot of us have been spending our lives for the last 18 months or so. But certainly want to make sure that if there is anyone new, they understand the features. If you're on a computer, the best way to do this is just sort of hover your mouse over the Zoom screen, and it should open up some options or a panel on the bottom. It's possible to change the location of that, so it could be somewhere else, but it should open up some panel. And you'll see the option to for a Q&A. And the Q&A is where we hope the feature that we hope you'll use if you have questions that you hope the panelists will address during a question and answer session, or when we have a bit of a break. And those can be questions that you feel like everyone might want to know the answer to, or just something that you want to know the answer to. And we might type an answer in there, or we may choose to answer that question live, depending on its nature and time and all of those other things. But we will try to address the questions as they come in. You will also find a chat button down there. If you click the chat button, it'll give you the option to chat with all of the panelists. I believe you should also have the option to chat with all of the panelists and other attendees. This is a place where you can chat, comment, connect with other attendees directly. I believe you should have that option. You can just click on the little gray box above the type message box, and you should be able to choose who you're sending those messages to. The panelists here will also be using that chat box to be sharing links with you and other resources throughout the course of the webinar today. So we might, share a link to a page that we're talking about, or resources that we think you might find useful sort of beyond the webinar today, or something that you may return to after the webinar is over. I think those are the key features that we're going to talk about today. There's also I think you'll see a raise hand feature there. I'm just going to speak for myself here and say I would appreciate it if you did not use that feature during the webinar, at least while we're presenting. It at least on my screen, creates like an orange flash while I'm presenting, and I just find it to be a little bit distracting. If you want to raise your hand during a break or during a question and answer session and potentially speak your question, we can certainly make that an option. I would just ask that you not do it while we're actually in the middle of a presentation, because I find it to be a little bit distracting. The webinar today is being recorded, and we will post the modules either individually or as an entire unit, probably within 2 weeks of the end of this webinar. So you'll be able to return to it and view it as you see fit. So I think that's about it. I'm gonna let everybody introduce themselves now, and let's start with Hilary, and then we'll go to Jeff, and then we'll go to Leif, and then Leif, when you're done introducing yourself, you can feel free to just jump right in and start with module 1. Thanks again for joining us, everybody. A reminder, in case you've joined just a second or two late, my name is Rich Hatfield. I'm a senior conservation biologist for the Xerces Society. I direct our bumblebee conservation programs under the direction of our endangered species coordinator, Serena Jepson, and really happy to be here today and really excited about this cross conservation organization and agency partnership we have to help find Franklin's bumblebee. So thanks everyone for being here today, and thanks to the panelists for joining us.
Thanks, Rich. I'm Hilary Sardinius. I am the pollinator coordinator with the California Department of Fish and Wildlife and our Wildlife Diversity Program. And we are doing a lot of work partnering with the Xerces Society to try and better understand the distribution of bumblebees, especially at-risk bumblebees in the state of California.
Good afternoon, everybody. My name is Jeff Everett. I'm a wildlife biologist with the U.S. Fish and Wildlife Service here in Portland, Oregon. I'm our agency's lead for Franklin's Bumblebee and the co-lead for Western Bumblebee. And I've been working on a wide variety of bumblebee projects with many of the people on this webinar over the last couple of years, and excited to show you how the partnership has grown and all the good work that's happening. So thank you, echoing what Rich said, everybody, for being here today.
And I'm Richardson. I work for the Xerces Society, although I've only been here for about 3 months. Previously, I was working as an environmental consultant and various other sorts of academic-based jobs. My research focuses on bumblebees and other bees and pollination ecology. And at Xerces, and with the colleagues on the call, I'm leading the California Bumblebee Atlas, which is beginning this year. So with that, I will share my screen with you. We will start the first of 3 modules that we are going to present to you today. Let's see here. Okay. Can I hear from a panelist that you can see my slides?
Yep, we're seeing the presentation, Lee. Thanks.
Okay. Well, welcome to Finding Franklins. Personally, I'm very excited about this webinar. I think it's a really important conservation undertaking, and I'm really glad we have so many folks who are interested in working with us on this. So in this first module, I will talk about some background about bees and bumblebees, the natural history and ecology of these animals, and this will set up some of the material on franklins that I'll present later in this first module, and that the rest of us will be presenting further on. So I just want to reference the authors here, Rich, Leif, Jeff, and Hilary. You have met, and we're also including Laura Tessler from Oregon Department of Fish and Wildlife on this slide. And we're really happy to have support from a diverse group of institutions on the right side of this banner photo here. And so thank you to all who have contributed to this effort. All right. So why, sorry, slide. There we go. So why do we care about, excuse me, everyone, I'm having a hard time seeing the slide under the Zoom stuff. First of all, I want to talk to you about a little bit about the Xerces Society and what we do. So we're the Xerces Society for Invertebrate Conservation, and we protect invertebrate wildlife through the conservation of the individual animals and their habitats. We divide our work among 5 different focal areas: native pollinators, endangered species work, work on aquatic invertebrates, butterfly conservation, and pesticides. We conduct education, conservation research, and advocacy to protect invertebrates and their habitats. So we do this through direct interactions with volunteers and other stakeholders, such as yourselves today, through field research and through publication. We work with public agencies and private landowners to conserve invertebrates and other species. And our work is in this work, we are doing science-based research. So we do an awful lot of community science, sometimes called citizen science. This is an example of one of those initiatives, right? So we've got this project, we're going to train you, give you information, and we hope that you will be interested to join us to do some field research to try to find Franklin's bumblebee. But we have many other community science projects, including Bumblebee Watch, Thanksgiving Monarch Counts, Pond Watch, Western Milkweed Mapper, and others. We do an awful lot of policy work and advocacy. And so our employees will testify before state or federal representatives. We try to advance legislation that may protect invertebrates and their habitats. And we do lots of education and training. organization is about 50 years old. And just since 2008, we've trained more than 100,000 professionals in the type of work that we do. And the geographic distribution of where we do this work, it's all 50 states, as well as Canada, Mexico, and off the continent in Europe and Asia as well. So we do a lot of applied science to actually conserve habitat and species on the ground. So we've supported pollinator habitat restoration on over 900,000 acres of land, and we've protected and restored over 1.5 million acres of land for rare and at-risk invertebrates. So this is really important to marry the sort of the community science, the scientific research that we do with and without those volunteer partners, and other aspects of our work with actual on-the-ground restoration and habitat creation. We're a nonprofit, and thus donors make it possible. And so we are just making this statement and showing how we can how people can donate to Xerces if they're interested to do so. So here's a quick outline of the 3 modules that we'll be presenting to you this afternoon. So the first one is about 50 to 60 minutes. It's, as I said, about bee and bumblebee ecology, natural history, and other aspects of how they live. We'll have a break, and then we will present module 2, which is shorter, 30 minutes. And Rich and I will present this module about how to find Franklin's bumblebee. And then in the 3rd module, Rich, Jeff, and Hilary will present survey protocols and identification, and some other aspects of that. So we can begin module 1, where I will talk to you about the ecology of these animals. And this is a photo of Bombus franklini. This is, I want to, with this photo, just make the point that we have very few actual photos of this animal in a live state. I don't know, fewer than 10, maybe 15 at the most, photos of that people have taken of a live Franklin's bumblebee on a flower. This is Franklin's bumblebee, but unfortunately, it's a specimen that was posed on a flower for a publication. So just to give you a sense that images of this animal going about its business are actually quite rare. And so broadly speaking, here at Xerces and with our partners, we're interested in conserving invertebrates. And so some will ask, well, why invertebrates? Why are they so important? And so we have this nice quote: "The fate of the world's insects is inseparable from our own." And insects and other invertebrates perform a wide array of functions in the ecosystem that without which we would not enjoy the same sort of life and lifestyle that we do. So for example, decomposition, you see a picture of a dung beetle there, biocontrol, so natural enemies of I think that's probably an aphid in that image, second to the left, are very important. So there's native species that might come to farms and parasitize the pests of the crop plants. This is something we don't pay for. It just is a benefit that comes to us from nature in most cases, and it's very important to agriculture. Invertebrates are often the base of food chains, so they serve as the food for larger organisms and then larger above them and so on. And most important to what we're talking about today, pollination. So bees, and especially bumblebees, are very important pollinators of both crop plants and wild plants. So we like to include this slide just so everyone's on the same page. Virtually everyone sort of knows what pollination means, but technically speaking, it is just one thing. It is the movement of male gametes from the male parts of flowers to the female parts of flowers that results in eventually in fertilization of ovules and the production of seeds and fruits. So many plants accomplish this in a passive way through wind transfer of pollen between flowers, but bees and other insects and other vertebrate animals are necessary for pollination of many species. So research shows that something like 85% of angiosperms of flowering plants require some animal for to be pollinated and to reproduce, at least in part. So that I'm not saying that 85% are wholly dependent on animals, but they're at least in part dependent on animals. And many of those are, of course, wholly dependent on a bee or butterfly, some partner to move that pollen between flowers. Now, pollinators are really important to crop production, as I said, and we know that that bee pollination accounts for something like 10% of the total economic value of agriculture. And so, I guess I should say pollination, not just bee pollination there. But if you think about the value of US agriculture, for example, fully 10% of all of that production is due to the fact that we have bees and other insects moving pollen between flowers in the crop fields. And this is importantly done by honeybees, which are a non-native agricultural animal. But studies have shown that in many cropping systems, honeybees' benefits to crops are outweighed by the benefits brought into the farm from wild bees and other insects. So taken as a group, wild bees are very important to crop pollination and to our diet, but also the economic value that redounds to agriculture from the environment. So here's a visual comparison of what our diets might look like with pollination by bees versus without. And so this is the first picture where bees were involved. And you see a number of pollinator dependent crops here, apples, citrus fruits, avocados, tomatoes. And if we lost bees and other pollinators, this is something like what the produce section of your grocery store might look like. So there are a few orange or other citrus varieties that are self-pollinating, but some of them are not. And so those are removed from the table. We've removed apples, we've removed a number of other things. So it's important to understand or to state that there's plenty of food for us to eat just with the wind pollinated plants, but we lose lots of the variety in our diet, lots of the nutrition in our diet if we don't have pollination. So this is an important ecosystem service when we're talking about wild bees pollinating crops. And I want to extend further, it's important to understand that pollination is a critical function in the ecosystem. And so the pollination of wild crops by bees and other insects results in the production of fruits and seeds that vertebrate wildlife species depend on, things like songbirds, right up to bears. So pollinators are functionally important to the structuring of ecosystems in that they help plants reproduce. So here are the most important groups of insect pollinators. We have a butterfly, a fly, I think that's a moth on the top right, a wasp, beetles, and then the bee on the bottom right-hand corner. And I've highlighted the bee to just make the point that while these other groups of insects are very important as pollinators in nature as well as in agricultural situations, by far and away, the most important group of insects for pollination is bees. And you'll see a bit soon why that is. And it has to do with the diet of bees. So one thing that distinguishes bees from all of their closest relatives, which would include things like sawflies and wasps and ants, is that bees are vegetarians. They collect pollen from flowers to feed to their offspring. And that's to be contrasted with, let's say, a wasp that might collect the larva of some Lepidoptera and feed those larvae to their offspring, or a wasp that might collect spiders and feed those to its offspring. So this is a sort of evolutionary novelty that took place millions of years ago that allowed bees to exploit a new protein source, plants. And we saw a rapid expansion at that time in the number of species of bees, as well as the number of species of flowering plants. So the reason bees are so important as pollinators is that they are actually dependent on flowers, whereas the wasp in my example might be gathering nectar from a flower, and that's important to the wasp, but it's not fundamental in the sense of being the protein that is necessary for reproduction. Okay, so what goes along with that is that bees have a number of adaptations for collecting and carrying pollen home and processing pollen in various ways. And I have a note on this slide just to remind you that pollination is incidental when bees perform it. So they're not doing this by intention. They're moving between flowers, foraging out of self-interest. And as they do so, it's a bit messy and pollen moves from male to female parts. there are a few pollination systems where the pollinator is actively moving pollen and placing it on the stigma, but that is not the case with any bees that we know of. So it's not just out of goodwill that they're doing this, it's their self-interest in getting a meal. So in this case, this bee is a relative of bumblebees. Melisodes dentroventus is in the same family of bees, the Apidae. And this bee has, for carrying pollen, has what we call a scopa, which is this hairy area on the hind leg on a part called the basitarsus. And you can see in this blown up image that this scopa is composed of many branched hairs, this thicket of hairs, and electricity holds those pollen grains in there. And sometimes the bee mixes nectar in and makes a sort of a goo that can stick to the hairs. Here's a bumblebee with a different type of scopa. Now, in this case, you can see that bumblebees have, rather than that thick brush of branched hairs, they have a shiny, concave, hairless area in the same place. And then they have stiff bristles that arch over the top of that hairless area. So this is a female Bombus terricola. Bombus terricola is a close relative of Bombus franklini, Franklin's bumblebee. So we can talk more about this one later, perhaps. And I actually have an image of Bombus franklini with pollen in that scopa. So I've circled the pollen load there. And again, the pollen is sitting on a hairless area, and then there are hairs that are sort of bristles that are over arching that pollen area and sticking into it a little bit. All right. So in addition, many bees are important as pollinators because they're specialized on their host plants. So just as you may be familiar with a Lepidoptera, like a monarch butterfly, being a host plant specialist, in the sense that the only place that the mothers can lay the eggs to produce more monarch butterflies is on plants in the genus Asclepias, milkweed, right? It's a specialist. They actually have to have the cardinalide toxins that are in the milkweed as a part of their defense against parasites and predators, right? So with bees, we see an analogous situation where many species of bees use just one or a few different types of plants for their pollen, even if they would use other plants for nectar or perhaps other resources. And so this means that numerous types of bees have adaptations that are specific to those types of plants. They may be a sort of a brush that helps them to pick up the pollen, or some other sort of morphological adaptation that's required to harvest the particular pollens of that plant. And this tends to make those bees very effective as pollinators of those plants. Bees are central place foragers, to use a wonky term from 20th century ecology. It just is a way of saying that they have a place that they live, and then their foraging takes place in a radius around that area, right? As opposed to a bear that is moving through the landscape, maybe has a territory and is returning to the same places, but is not coming back to the same central location after each foraging bout. And this has, I just raised this because it has consequences for the plants and the bees involved in that interaction. And lest we forget, aside from pollen and nectar, bees are collecting a number of other things from flowers. Not so much bumblebees, but other types of bees are collecting oils that are secreted by flowers, sometimes resins or volatile chemicals for mate attraction. So there's all sorts of other stuff that goes on in the interaction between bees and their pollinators. So now I'm going to present a series of slides to just show you some of the diversity in bees, and to convince you of the numeric diversity, species richness, as well as the sort of diversity of form and function that we see in nature for these animals. So globally, we have more than 20,000 described species of bees. We're confident that there will be more. In fact, it's a common thing to see a new bee described in the peer-reviewed literature. And so that number steadily creeps up. In North America, we have about 5,200 bee species. These are the native species that occur here. And then just in the United States and Canada alone, that's 3,600 species. So dropping off the species that are in Mexico. And I will present some stats on just that last statistic now. This is a nice graphic that was made by a colleague named Joel Gardner at the University of Minnesota. It may be a little bit difficult to read, but it breaks up the diversity of those US and Canadian bee species by their genera. So top left in orange, you can see bumblebees, and it says the number 50. And so the point there is that there are 50 bumblebee species found across North America. Right next to that, you see honeybees and just one. There's only one species of honeybee in the US and Canada. It's the Western honeybee, sometimes called the European honeybee. As I said earlier, it's not native. It's a farm animal. It's also a naturalized non-native species, or you could say an invasive species in some cases, where it lives without the aid of people in suitable habitats. So if you go around that pie chart, you see there's an awful lot of diversity in some of these groups of bees that you may not have ever heard of or know very much about. I'm certain there are people in the audience for whom this information is not news, but I just want to point out there's an awful lot of diversity among some small, fast-flying, ant-like sort of bees that most folks would just overlook at flowers. They're going by really quickly, and they're quite small, or they look a lot like a wasp or something else. And also, I just want you to see how few of these bees are bumblebees. Only about 1.5% of all the bees here are bumblebees. And some other things that make bumblebees a little bit unusual. Most of the bees in this figure are solitary, meaning that they, the mother, after mating with the male, provisions a nest, lays eggs, seals up the nest, and after that, the male and the female adults die at some point, and later, maybe the next year or the next season, the offspring emerge. They never meet their mother. There's no social interaction between generations, or usually between individuals of the same generation, other than that mating interaction. By contrast, bumblebees, like honeybees, are social. There's a caste differentiation and all sorts of behavior that is necessary to maintain the collective that they live in. And then I finally just want to make the point that that about 65% of these bees are nest underground, and most of them dig a hole. They use their forelegs and their jaws to move dirt backwards behind them. They can dig quite deep, lots of labyrinthine sort of chambers and tunnels. And then about 35% nest above ground in preformed cavities, or perhaps for a few of them, they actually dig their own cavities in wood. Bumblebees are a little different in that there are some species that nest above ground and some that nest below, but they generally cannot create the cavity that they nest in. So they have to find a cavity that has already been excavated by a rodent, or it's a dry, dark spot in the back of a shed, or it's some sort of natural cavity in a tree. These are suitable bumblebee nest sites for some species. So I'll go through a series of slides that just show representatives of some of those families of bees. First of all, here's the Western honeybee, Apis mellifera. So again, the family is Apidae, which is the same family as bumblebees. They are highly social, much more so than bumblebees, in that they are a close relative of bumblebees. Here we see the largest known bee on Earth, and I believe that's the smallest known bee on Earth. The big one is Megachile pluto. It is a bee from Southeast Asia that is, I don't know, it's 2 to 3 inches long, I think. Whereas the other one is a Perdita species. It's native to North America. And you can see just how small it is related to this much larger bee. Here are 2 examples from the family Halictidae, or the sweat bee family. The one on the left is in the genus Halictus. The other, we often call these green bees or metallic green bees. This genus of green bees is Agapostemon. So these bees look quite different. They're relatives and have similar, broadly speaking, similar ecology. Here's one more example of another genus of green, metallic green halictid or sweat bees. Photo by Clay, who is in the audience, I saw. This is a species of Andrena. This is a species-rich group in North America. Many different types, quite a a range in size, specialization on plants, very important pollinators found coast to coast. This is a representative from the Megachilidae. It's a leafcutter bee, and the genus is Megachile. These bees are just fascinating in that they incorporate leaves and mud and sometimes other materials into their nest architecture. And so they actually go out and forage for leaf pieces. And here's a female who's carrying home a piece of leaf that she's cut off of a live plant. If you get the chance to watch this happen, it's just marvelous to see them use their mandibles to cut a piece of leaf, a nice little circular piece, and take it home. They mash that up. They may combine it, depending on the species, with clay or other mud or something like this. And they use it to make plugs that separate the different cells in the nest and sometimes other parts of the nest architecture. This is a cuckoo bee. It's in the genus Nomada. I believe these are apidae, just like bumblebees. This doesn't look anything like a bumblebee. This is a parasite. We call it a kleptoparasite of the genus Andrena that I showed you previously, as well as some others. This bee is somewhat wasp-like, right? It doesn't have a whole lot of hair. It hangs out around the nest entrances of its hosts. And when the female leaves to forage, it sneaks in, klepto meaning, I think, thief or to steal, right? Sneaks in, lays an egg near the pollen that the mother has provisioned her own egg with, sometimes in the wall of the cell where the egg and the pollen are. And then the offspring of this bee emerges and consumes the pollen and perhaps also the offspring of the host. So there are a number of parasitic species, socially parasitic or kleptoparasitic species in the bees. And finally, here's a bumblebee. This is Sanderson's bumblebee. And you can see the bumblebees are densely hairy. They tend to be larger than other types of bees that we've been talking about. And one more, and this is another type of bumblebee. This one's a male, and we'll talk in depth about how to distinguish males from females, like the last slide, a little bit later this afternoon. All right, so to wrap up that diversity section, I wanted to show you a map of North America showing bumblebee species richness in these arbitrary grid cells. This is from our 2014 Field Guide to the Bumblebees of North America. We have better data now, but I thought I'd show this one. And you can see that the range of Franklin's bumblebee is an area with some of the highest bumblebee diversity anywhere in North America. And the presence of Bombus franklini is one of the reasons, it picks up by one species in that area. But just more generally speaking, this is a really important area for bumblebee diversity. So not only do we have Franklin's bumblebee, and as you'll see, the range of that species is very small, but we have a series of species that are Pacific coastal endemics, you might say. They don't make it very far inland, but some of them do occur with quite a good latitudinal amplitude from somewhere in Central California all the way to Southern Alaska, for example. And you've seen this. I'm going to skip right by this again. And now I'm going to talk to you about bumblebee ecology, natural history, ecology, how they live, so we get a sense of what's going on for Franklin's bumblebee when it's underground, when it forms colonies in springtime, and so on. So there are about 50 species of bumblebees in North America. As you've heard previously, this number changes as we see species divided into a pair of sister taxa, or in rare instances, somebody discovers a new species, a cryptic species that was there all along that we never recognized as distinct from its sister species, for example. So we have about 50 now. That number will creep up slowly a little bit, I would guess. Bumblebees are social, as I said before, just like their relatives, the honeybees. What's different is that they form colonies that are annuals. They start in the spring or the beginning of the growing season, and in nearly all cases, that colony finishes in the late summer or fall with the production of reproductive offspring, and then the colony dies. Everybody dies except for the new female reproductives who have mated, and they hibernate and start the new colonies the next year. By contrast with honeybees, you have colonies that can last for multiple years, and the same queen can live for multiple years also. So there's more of a continuity there. Bumblebees are also less highly social than honeybees. They are social, and we can talk in detail about the differences, but they lack some of the behaviors you may have heard about for honeybees, such as the waggle dance that allows honeybees to transmit information about the distance and the direction to food resources. That's not something that we know that bumblebees can do. So as I said before, bumblebees often nest underground in abandoned rodent burrows. They can nest on the surface of the ground in bunch grasses. I found them in sedge hummocks in wetlands about an inch above the waterline. All sorts of on the ground sorts of situations, providing that they have some cover. They're not uncommon in a rock wall, in the wall of a house where you'd expect insulation, sometimes in the rubble around foundations of buildings. Bumblebees seem very flexible in their nest site choices. There aren't too many species where we say the only place they can nest is X or Y. We see some variability in most of the species. And the fact that they use anthropogenic or human-created habitats at times, like walls of sheds or that sort of thing, makes us wonder what's the reference habitat? What are they really looking for? Anyway, it doesn't seem like it's a highly specialized resource need, but I should note that some researchers believe that nest site abundance is limiting to bumblebee populations. And so as far back as Charles Darwin, who was a bumblebee researcher, we have people speculating that the rodent populations in a particular region are really important to understanding the bumblebee populations because of their limiting factor on nest site selection. So we're not certain that that's accurate, but people have been speculating about that for centuries. So here's a look at the annual cycle of a bumblebee colony. So starting in spring, quote unquote spring, well, we can say spring in the range of franklini, right? The females, the reproductive females who have mated the previous year, when they emerged from their colonies, they're known as gynes, G-Y-N-E. You can call them a queen if you want. When they hibernate and then they come out the next year, we call them queens or foundresses. They emerge in spring. They spend several weeks foraging for resources and looking for that nest site. Once they've found a suitable nest site, they start to return pollen and nectar to that site. They make a waxen sort of cup. And to do this, they use wax that they secrete from glands on the underside of their abdomen. They fill that with nectar and pollen, a mix of the two. They lay eggs on top of it, which are encased themselves in wax, and then they incubate. They're warm-blooded partially, and so they can shunt heat to the abdomen, heat up those eggs, and they just sit right on top, just like a bird. It's pretty interesting to watch, and transfer heat right to their offspring. As I've said, in some cases, we think there is competition for nest sites. And one line of evidence for this is something we call super seizure, where in some cases, if you are lucky enough to find a bumblebee nest as it's getting established, you may find some dead queens outside the front door. And in that case, we would interpret those to have been other queens that came in and tried to take over, or it's the original queen who was killed by some interloper who took over her nest site. So it is possible that queens are fighting over these sites, and the nest site quality is some index of the fitness or reproductive potential of that colony. Maintaining the thermal environment is critical for bumblebees. So insulation is often part of that picture, as is bees' behavior. So they will fan at the entrance with their wings to move air out, or sometimes in, I guess. And so when we stick a thermometer in a bee nest, we find that they're maintaining a very stable and fairly high temperature at all times around the larvae and the eggs. And that's the same for humidity. They manage humidity to avoid the growth of fungi and other microbes that they might not want. All right. So moving on to summer. So I left you with the queen incubating those eggs, right? And on the left-hand side here, we see some things that look like rice, and those are the eggs, those elongated white things. And they're inside of the wax cup that has been broken apart so you can see the eggs a little bit. Those eggs will hatch after a few days and become larvae. And then in the next picture, you see an adult bee tending those larvae. So the workers, first the queen, but then later the workers of the colony, which are non-reproductive females, will get pollen and nectar, and they'll open up that wax cup and they will feed each bee and each larva individually. There's some diversity in how bumblebees accomplish this, but that's a general truism. And as the larvae pass through several different stages, the workers and or queen will move them to larger wax cups. You can see that they're collective, they're in the one together at the beginning, but later when they get larger, they get their own waxen shell. And when they're ready, they pupate. So they go into their final stage where they transform from this big bag of juice, this larva that looks like a grub. And over the course of 5 to 10 days, they transform into the adult bee and emerge. So as I said, there is the queen who founds the colony. There are non-reproductive female offspring, which we call workers. And that's all that the queen produces at first. She produces usually fewer than 10 eggs in the first clutch, and they all become non-reproductive females. And then those bees take on almost all of the work tasks of the colony. They forage, they feed the queen, they clean, they do everything for the maintenance of the nest. And this cycle repeats until the colony, quote unquote, decides it is time to reproduce. And we can talk in detail about what that means if people are interested. But for now, I'll just say at some point, the queen stops laying eggs that are destined to become workers. And now she lays eggs that are raised to be reproductive females, queens or gines, as well as drones or males. And eventually the colony, depending on the species, can get up to over 1,000 individuals. However, this is not typical. And most of us who work on these animals are familiar with colonies that might contain 100 animals or maybe 200, but there are definitely species where the colony never gets bigger than 50 to 75 bees. So you can contrast that with a honeybee hive where you may have 10 to 20,000 bees at the outset of the summer, and by the end of the summer, you can have 50,000 or more in one colony. Just one queen, and everybody else, virtually everybody else, is a worker. In the fall, after reproduction, the reproductive females, the gines, they go out, they find males, they mate, they feed a great deal on fall flowering plants. So I'll lay this down as a marker that flowers are necessary in fall for bumblebee colonies to succeed. We need the queens to have something to eat because they need to fatten up and have stored resources internally for the long winter or diapause that they're going to go through. They then look for a place to do that. Typically, bumblebees dig. This is a time when they do excavate their own hole. However, they dig only about 2 inches down, just below the root line in a grassland, for example. In this picture, maybe 2 inches below those grasses, and then just bury themselves. And so they're spending the winter not in an open subterranean area, but just basically buried with little airspace around them. And they go into a hibernation or a diapause there. They often will use north-facing slopes in forests, maybe under trees or other sorts of easy to dig substances like rotting wood or in the leaf litter. But I should say that for most bumblebee species, including Franklin's bumblebee, we really do not know enough about this process or this compartment of life history to be able to manage that habitat or to use that information to help recover a species. So we just don't know where Franklin's bumblebee goes in the winter. We assume it would be underground like this, but I'm not aware of any records of that. So here's the cycle in this beautiful image illustration by Alex Lucas. And you read it from left to right. Left is winter, right is fall. And you can see in this case, the example is a rusty patch bumblebee. You probably know this one is federally listed as endangered, and its habitat is in the east from southern Canada in Ontario, east as far as New Brunswick, and then south in the mountains as far as Georgia. And it's most common now in the upper Midwest, in states like Minnesota, Illinois, Michigan, Iowa, and in Illinois. I said Illinois. And I want to say also that the rusty patch bumblebee is another of the relatives, the closer relatives of Franklin's. So I think it's worth paying attention to the closer relatives when we think about the data gaps, the information gaps for Franklin's bumblebee, even though we're talking here about an Eastern US, Eastern North American species. So, in the red box at left, we have the queen emerging from her hibernation in springtime, finding flowers and a nest site and initiating that nest. And you can see in the bottom of that red box, that queen is in a pose that is very typical for incubation. They lay over the top of the wax mass with the eggs inside, and they spread out their abdomen so that they can transfer as much heat as possible. Later in the summer, we have lots of workers in the colony, and the colony can be said to peak in its numbers. There's a fascinating natural history where the queen and the workers have to work or compete to figure out when this switch happens to reproduction. And I'm not going to go into it now, but suffice to say, there's a lot of behavior and ecology in that step that determines the fitness of the colony, the reproductive output. So sometime in summer or fall, those queens or gines and males are produced. They find each other and mate, and the queen, after feeding, goes to hibernation, and the cycle repeats. We want to make you aware, if you're not, that there is a special sort of interloper in this situation, which is the cuckoo bumblebees. So it is a bumblebee. It's the genus Bombus, and they're not more primitive than other bumblebees. They're not the closest relatives of bumblebees, as people thought for a long time. They are actually bumblebees embedded in the family tree of bumblebees. However, they're just amazing. They've lost, because they're parasites and they attack the colonies of other host bumblebee species, they have lost lots of the adaptations that we're going to tell you that Franklin's bumblebee and other bumblebees have for things like pollen carrying and the behaviors necessary to keep a colony cohesive. So typically they will invade the nest after the queen has started to produce workers, but before there are too many workers. This female cuckoo bumblebee will then control the colony through a combination of aggression and pheromones, eventually killing and eating all of the resident eggs and larvae, and sometimes killing the queen herself, and then laying her own eggs and forcing through aggression and pheromones the workforce to rear those offspring. So after this attack, if it's successful, the only thing that comes out of these colonies besides foraging workers is the offspring of the attacker, of the cuckoo bumblebee. And so as I guess you'll hear later, I don't believe we know of any cuckoo bumblebee that attacks Franklin's bumblebee. It's entirely possible that there are some, but we just don't have enough information about this particular rare species to answer that question. Sorry, and you may be aware of Bombus sucklei, or Suckley's bumblebee. It's a species of conservation concern throughout the West. It occurs, its extreme southern range margin on the West Coast is Northern California and Southern Oregon. So same geographic area we're talking about today. That is a cuckoo bumblebee, Bombus sucklei. So that gives you a little bit of a sense of one of the players here. So where are we going to go to find bumblebees? What sorts of habitats? Here's some data from the Pacific Northwest Bumblebee Atlas, which is an ongoing project of the Xerces Society and our partners. And this data is just showing you the percent cover of different land cover types around survey areas and around collections of individual bumblebee species. And so, well, actually all the species put together. And so I just want you to cast your eye down the list of words and see that we catch an awful lot of bumblebees in grasslands or meadows. That shouldn't be surprising because we often catch bees at flowers, and open areas like that tend to have higher floral density than maybe a closed canopy forest. Forests, however, are very important for bumblebees, some species of bumblebees. We often find bumblebees in developed areas, as I referenced before. And then these other sorts of habitats, wetlands, riparian areas can be really important for the plants, the flowering plants that they produce, as can shrub scrub areas. And we do find that bumblebees in agriculture, as I said, they're important pollinators. And there are some species that seem to depend on agricultural type habitats for part of their life history. This is from the same project, it's the same data, now broken out by species. And we have this messy plot just to show you the truisms or generalities that I presented in the previous slide are just that, they're generalities. And so we see a lot of variation among the species in the types of landscapes or cover that they tend to occur in. So at the top, Bombus vosnesenskii, a very common one that you're probably familiar with, is often found around in developed areas. Also found in open areas like grasslands. By contrast, Bombus van Dijkii, just below that one, very different set of habitat types, and it's especially found in woodland and forests in the Pacific Northwest. So we need to think about each of these species as having their own adaptations to using the environment to extract resources and reproduce. So a couple of slides about what we don't know about bumblebees generally, and this should stimulate your thinking about how to approach some of the questions with in terms of finding franklini. So about overwintering, there is limited evidence. We have certain species where we have lots of documentation. In many cases, it's just anecdotal. We have this paper or person saying this thing, and then there are a few other observations, but you can't draw very many generalities from just a few anecdotes. And for many species, we just don't have any observations of how they do this. And as I said, franklin's is one of those species. So generally speaking, overwintering takes place underground in these shallow burrows, but that is me saying that based on what we know about common species, right? So it's entirely possible that there's more diversity to the way that the bees spend the winter, and that we should get to know them better. It's important to understand just how much time they spend in diapause. In temperate zones, this can be more than half of the life cycle. It can be 9-10 months even in northern Canada, in the subarctic. So what happens underground during that period of relative immobility and lack of activity is very important to bumblebee persistence. And here's a place where we humans just don't yet know enough to help them along, right? So we want you to see this, this link here to queenquest.org, which is a recently started initiative to find queens or gines going to hibernation, or that have just recently gone into hibernation. If you're interested, this is another community science project that is a really fun one, trying to solve these riddles together. And I would encourage you to follow that link and see if you would be interested in participating. Or please get in touch with one of the presenters if you want more information. I should have clicked this already. This is a video. And here you see a queen working on what we think is probably a hibernation site. So she is digging in the depth there, pulling dirt and pine needles and other stuff out of that hole, and using probably all 3 sets of legs to move that stuff backwards. Eventually, she'll go down into the tunnel, pull everything in on top of herself, and that's that. I've watched this. It's very fascinating. So some uncertainty centered around nesting. So again, we have data from common species, but we lack data from most species about just exactly how they reproduce, what goes on in the nest, how specialized are they in terms of using, say, a chipmunk burrow versus some other sorts of suitable nest sites. We know very little about how to manage for bumblebee nests. So for example, there have been, because we have a bumblebee on the federal endangered species list, there are cases where development has been questioned or studied before the actions were taken to try to understand impacts on nests. And I would say that we end up in a place of uncertainty, saying, well, we have a problem. We're not sure if we're going to squish nests with this development project or not, because we can't even really tell you exactly where those nests are or how dense they are, how densely packed they are in an area. So again, this is a place where we need more information. And Bumblebee Watch, which is one of Xerces community science projects, it's our online portal for data, Bumblebee data entry and storage and curation, is a place that you can report nest sightings. So if you make an account, you can do the normal thing, which is to report a live bee flying or foraging, but there's also a way to report nests. So we really would like to have more information about nests. So please get in touch if you find one. Again, I neglected to show you the video. So here you see a queen and a worker. I believe that's Bombus and Patience, the common eastern bumblebee. And there's a nest hole there that you saw the big bee come out of, and now we see the little one go in. Okay, so it's pretty hard to find them if you go looking. They're very good at disguising the entrance and entering and exiting in surreptitious ways. Okay, so a few other issues about what we don't know. We know something about how far they can fly to forage through some really creative experiments using marking as well as some genetic methods. But we don't know that much about how much they disperse and what those barriers to dispersal might be. So after production of those reproductive offspring in the summer or fall, do they fly 100 kilometers away? Do they fly 5 kilometers away or even less? Do they stay local? For many species, we just can't say. Related to that, it's hard for us as the scientists to say what is a healthy population density for these guys. How many nests do you need per unit area or on a landscape, let's say, to know that you have a sustainable population of a species of bumblebee where there's enough genetic diversity and so on to ensure persistence. There are some things about bumblebee reproduction that are not well understood. Courtship and finding each other. There's some interesting stuff about males and sperm competence, about sperm storage by females over the wintertime, and pathogens that might intervene in reproduction. So we need more information about that. And relative to what I just said, we in general study female bumblebees when we study bumblebees, and the males are important, everybody knows, but we don't have a lot of really great studies of some of the aspects of life history that are specific to that sex as opposed to the females. So they're not as important, frankly, in terms of the fitness of the colony, but they're obviously critical to the fitness of the colony. And we should do better at understanding the ecology of male bumblebees. So the value of bumblebees, as I've made the case, they are really important as pollinators. This is another one of those cuckoo bumblebees foraging at a flower. So they feed themselves, they just don't carry pollen home to feed their offspring. Bumblebees are particularly important as pollinators due to the way that their form and sort of their function. So because they are warm-blooded, they are able to fly in cold and wet conditions. You can see them flying at about 35 degrees Fahrenheit, but they can also be foraging up to maybe in the 90s Fahrenheit. They're smart relative to some other insects, and they are capable of learning. In fact, it is a typical sort of thing to see from a nascent forager who ages over the course of her several-week lifespan. They go from one foraging task to progressively more difficult tasks, and they actually need to learn how to do some of this. So the two rightmost photos show a bean family plant and a gentian, and both of those are a little bit tricky for a bee to handle until she has figured out how they work. And the one on the right is the gentian, where the bee has it's a closed flower and she has to cram her way in there and figure out how to pull the petals apart. It's very ungainly at first and they're not very good at it, but they learn how to do it and they become just experts at it. So their capacity for learning and flexibility is part of why they're important pollinators. And then just generally speaking, bumblebees have longer tongues, which allows them the flexibility to reach nectar in deep pockets as well as shallower pockets. And that means that more plants can benefit, more types of plants can benefit from that one species of bee. So here is a list of some of the crops that bumblebees are known to be really important pollinators for. Tomatoes, peppers, blueberry, cranberry, clover, I would add alfalfa there, squash, melons, apples. Just so many other crops are reported as being pollinated by bumblebees. One of the reasons bumblebees are superior pollinators is that they can sonicate flowers, they can vibrate a flower, they can really shake a flower and get the pollen to rain loose off of the anthers. And indeed, many of these types of plants are adapted for this. So their pollen is secreted away in these anthers that are difficult to get the pollen out of. They have a sort of a hole at one end, and the bee can't reach inside the anther. It has to shake the anther to get that pollen out. And in fact, it's the specific amplitude or frequency of the vibration that for some species makes the pollen come out. So honeybees can't do any of this. They're very important pollinators, as I said earlier, but they do not sonicate or buzz these flowers. And so to finish this section, here's a slow motion picture of a bumblebee hanging on to probably a solanum flower and vibrating her flight muscles without moving her wings. And you can see the pollen just raining out of those banana-shaped anthers. And that's what we're talking about with buzz pollination. All right, so I have about 5 slides more to introduce you to Franklin's bumblebee, and then we will have time for questions and a short break before we move on to the second module. So first of all, here's a little history of what we know about Franklin's bumblebee. So the period of observation, the records that we have of people collecting the bee or observing it, it starts sometime before 1908, because the oldest specimens that we're aware of, there's no date attached to them except for the date of the person who identified it as Bombus franklini, and he wrote a little label that said 1908. So it must have been collected at least that year or earlier. And the last known collection or observation was in 2006. The species was formally described in 1917. And interestingly, the species was described from material from specimens that had erroneous label information. So in that paper, this species is said to occur in southern Arizona, when in fact it does not. Dr. Robbin Thorp, formerly of UC Davis, who has passed away, a colleague of ours, is the single person who is most important to raising awareness of the existence of and the ecology of Bombus franklini, as well as its conservation needs. So as he was transitioning into retirement, he continued to do annual surveys of the habitats in Southern Oregon, Northern California, where Bombus franklini occurred. And in the mid to late 1990s, he noticed that the bee was becoming much rarer, and he publicized this. And he began to carefully collect data every year. And here is a look at that data from the last decade of Robbin's collections. And you can see that that as time went on, we saw fewer and fewer bees in his annual collections, which involves similar survey efforts. So we can kind of compare each bar to its adjacent bars. The last single individual he saw was in 2006, and the observations go on beyond 2007, but we have no more observations since then. So you will see some people refer to franklini as possibly extinct. That is a true statement. However, personally, I remain optimistic that there are a lot of places where populations could be holding on that we simply haven't investigated yet. So it wasn't until 2010 that a petition to list Bombus franklini was submitted to the Fish and Wildlife Service. That was written by Dr. Thorpe. And the last date I wanted to make you aware of is just that in 2021, California considered listing this and 3 other bumblebee species as endangered. And that didn't happen. And there's lots to discuss about that, but not at this very moment. So here are some of the potential stressors and threats that we think may have contributed to the decline of this species. That would include pathogens. So for example, there's a pathogen that we think was accidentally introduced to the US from European bumblebees as a part of the commercial bumblebee industry that was starting up in the 1990s. And we think it may be responsible for the decline of this species and some of its close relatives, including the Western bumblebee and the rusty-patched bumblebee. Grazing is causing impacts to the land where this species occurs. Invasive plant species are changing some of the habitats and probably crowding out some of the host plants, the forage plants for Bombus franklini. We know we've had some habitat loss and fragmentation. And I think that would mostly be concentrated at the lower elevation of the range of this species. And since many of the higher elevation sites are protected, fire is certainly a factor in these landscapes. And then there's at least one example where franklini habitat was impounded, was flooded with water for human use. This map shows you the sum total of all known observations of this bee. The gray dots are all of the records up to the year 2000, and then the orange-red colored dots are those that are more recent than that. So we only have a very few number of recent observations. Most of the observations are much older. And so it's imperative that we resurvey some of these former sites where the bee occurred to see if it's still there. Now, we can take those points and create a model that asks, given the environment, the environmental conditions where this bee was collected and all the other Franklin's bumblebees were collected, what's the range of possibilities on the Earth for environments that are similar that might support this species? And that's called a species distribution model. And this is one that I created for Franklin's bumblebee. So based on those points and then a bunch of environmental and land cover predictor variables, we're able to create this probabilistic prediction of where the range is for this species. And no surprise, it's centered around where most of the points are. But if you look closely at this, you'll see some places that where we have no existing occurrences or historic occurrences, yet the model predicts high suitability. So the darker green areas are the more high suitability areas. And we're a little skeptical about that. For example, that dot where it says Crater Lake National Park. However, these models they're hypotheses. And if we trust the model in terms of what went into it, it might behoove us to go looking in some of these places where there's predicted suitability, habitat suitability, and we haven't yet found the bee there. This gives you a sense of the timing of activity for the bee. So clearly, if we are going to find it, we need to be in the right habitats at the right time. And this is all of those records of observations plotted according to the day of the year and constrained to just when we have data for that. So starting in early to mid-April, we see the first queens come out. Queens are, and then workers. So females are on the landscape straight through to late September. We don't see the emergence of males until July, and they really don't become common until sometime in late August and September. Now, this graph is a little funky. You have these troughs. I don't think the bee is really going away. It may reflect the shape of some of the two lines may reflect biases in the data from when we went collecting or didn't go collecting, that sort of thing. And you can see from the y-axis that we really don't have much data overall. So if we had more observations, we could probably fill out those gaps and get a much better sense of when to expect the different sexes in this case. I wanted to show a plot of elevation at each location where we found the bee. And this is not necessarily anything earth-shattering, but as you can see from these box plots, in California, the average elevation of a collection or observation of Bombus franklini is around 1,500 meters, whereas in Oregon, it's substantially lower, about 500 meters. So, in Oregon, as you'll see, we have observations from Ashland and other lower elevation areas, as well as some of the higher elevation protected areas. In California, nearly all of the observations are coming from higher elevation places in the Marble Mountain Wilderness and adjacent mountain ranges. 2 more slides, I think. This is all of the host plant information that we have for Franklin's bumblebee. These are the genera of plants that we have recorded this species being on, and the number of observations is just that. So more observations we interpret to mean this plant is more important to the bee. Although you can think of ways that this data is biased, right, based on where we go collecting and that sort of thing. But we know these plants are important. So California poppy, monordella or coyote mint, vetches, knapweeds, lupins, clovers, and we'll get into the common names of some of these others in a little bit. But that's sort of the universe of plants that we want to start looking on. We certainly don't want to not look at other plants, but those are the plants that the bee has shown us it is using. So quickly, here are some data gaps for Franklin's bumblebee. They're basically the same data gaps I talked about earlier for all bumblebees, but this is a poster child for data gaps in the bumblebees, right? So we don't know that much about the nesting biology. We know nothing about hibernation. I speculated that there may be a cuckoo bumblebee that attacks Franklin's, but we don't know that. So pathogens, parasites, pests, we don't know much. I don't think there's ever been a sample of the gut parasites or pathogens of this species. We don't even know exactly what the range is like. I mean, I think we're confident it occurs in the 60 mile by 40 mile rectangle or oval across the two states, but I don't think we'll be surprised if you find one that's outside of the currently known range just a little bit, right? And so the current status, is it extinct? Is it phenomenally rare? We just don't even know. We haven't seen one for 15 years. So please help us fill these gaps by locating Franklin's bumblebee. With that, I will wrap up the first module. And I think we are going to take a little bit of time for questions, and then we're going to have a break.
Thanks, Lee. That's great. There are a few questions here that I thought might be worth addressing. One, since we were just talking about Franklin's and just introduced it, it might be worth talking about first. David Colin is interested in theories as to why franklini had such a small range, and interested in sort of learning more about it. But if you knew of any references that you could share that discuss this, I'm not actually aware of anybody that's really sort of put a hypothesis out there about this, other than you know the uniqueness of the Cascades Askew ecosystem being just a sort of really unique, interesting place in terms of biodiversity generally. But I don't know any specific hypotheses about Franklin's bumblebee. Maybe you do.
No, I don't. Sorry. It is important to note that many bumblebee species have really large ranges. So Bombus californicus, also known as Bombus fervidus, occurs from northeastern or eastern Canada all the way to the Pacific coast, and in various parts farther south. That's not uncommon to have this sort of coast to coast type of range. And this is a very, very small range. I can think of one other species of bumblebee that's similar in its range, but no, as Rich says, that's a unique sort of ecology there, maybe some sort of period of isolation over time from its congeners, its close relatives, but I'm speculating. We really don't know much about that at all.
Great. Well, not great. I wish we knew more, but so be it for now. But hopefully, if we can rediscover this species, we can potentially answer some of those questions, or have the opportunity to, at least. Question a little bit further back, more generally about bumblebee ecology, Leaf. Just about the question that I think the reason that I'm interested in sort of answering this question live is it asks a question about genetic diversity in bumblebees that I thought might be worth just sort of addressing as we're talking about an endangered species, and just sort of briefly discussing some of the issues surrounding that. So the question is, the males mate with new queens from the same nest, or do they travel to different areas seeking mates from different nests? And then follow up is there a concern about genetic diversity within the same bee species?
Great questions. Yeah, so bumblebees are interesting in that, as far as we know, most of those that have been studied, they just mate once. So the gine goes out and finds a mate, she mates, and she's done. Many insects mate multiple times. There are some bumblebees where we know they have multiple males involved. But you end up with a colony the next year where there are just two parents, as opposed to in honeybees, a good contrast. The nuptial flight, as it's called for gines, involves multiple matings in the air with many males that are up there searching for mates. That results in a colony with all this genetic diversity. You have these different lineages, male lineages. They all have the same mom, but they have different dads. So to answer your first question, bumblebees, no, they don't mate in the nest with their siblings. As far as we understand it, they go out looking for mates that presumably come from other colonies. Now, this is really important because the answer to your second question is, yeah, we have a lot of concern about genetic diversity as a factor in bumblebee declines. So when populations get small, we have fewer individuals carrying the genetic material of the species. We have fewer different versions of each gene, right, in the population. And in fact, with bumblebees, as with other Hymenoptera, so ants and wasps and some other insects also, they have this interesting way of determining the sex of the offspring, where it has to do with the diversity of I'm not going to give you all the details unless you really want to hear them, but diversity at one particular locus on the genome. In populations with a lot of diversity, it's very unlikely you'll have a homozygous individual where you have both parents have the same copy of that gene. But in small populations, it becomes more and more likely that happens. And when a bee is homozygous like that, some things go haywire, and I'll leave it at that. So genetic diversity is a factor in declines. And in fact, as it decreases, we see this spiraling, we call it a vortex of extinction, where the rarer they get, the more likely they are to have problems with genetic-based problems with reproduction, and therefore they become rarer. So genetic diversity is a big part of this picture. And you can see from my remarks earlier that there are some data gaps in understanding, dispersal distances and how big populations are and that sort of thing.
One more question just popped up about foraging range. What do you know about the foraging range of bumblebees in general?
So the farthest known dispersal in a marking study where they painted bees, or they moved bees somewhere, they painted them and then they let them fly home, is around 10 kilometers. And that, I think, is for Bombus terrestris, which is a European or Eurasian species. Like just by a chance, it is another close relative. It's in the same subgenus as Franklin's. So 10 kilometers is we often state this as like the maximum distance they might fly to forage. However, there are some really elegant genetic-based studies that look at siblings. You collect a bunch of bees in one place and look at the number that are siblings, and you can figure out the number of colonies that are there, and you can start to work out how far they had to fly to get there. That information suggests that many species don't forage farther than about 1 kilometer to 2 kilometers at most from their nest. So pretty local. We generally would say maybe a kilometer, a diameter circle around a nest is about what you'd expect is the universe for that colony.
Yeah, and as I understand it, that 10-kilometer study, it wasn't necessarily a foraging bout per se, but an indication that bumblebees likely know large enough landscapes at a fairly large sort of landscape scale to navigate effectively, not meaning that they actually would fly that far to forage, but in their sort of navigational flight bouts when they emerge from the nest or from a new location, they can see enough to sort of follow navigational techniques within 10K. Is that your understanding as well, Lee?
It is. And if you think about it, flight is expensive, is energetically expensive. And so they can't fly forever to go to some bonanza resource without stopping to refuel themselves. And then they can only carry so much home. And the more they're carrying, the more they're spending or burning through just to get home. So there are some limitations on how far they can fly in terms of the energetics of the colony. It just doesn't make sense to go farther than that for many of them.
Great. No more open questions. If you asked a question in the Q&A, I typed a bunch of answers in there. If you're interested in what other people may be asking and what I've answered, you're welcome to peek in there and look at the answers. And I'm saying this to attendees, not necessarily to you, Leaf, although you should check it out too if you're interested. Before we move on to the next module, I believe that Laura Tessler from the Oregon Department of Fish and Wildlife has joined us, and I just wanted to give her a chance to introduce herself and say hi, if she's with us.
Hi there, I am with you. I'm sorry I was late. I was telling Rich I was taking a CPR class, so and learning a first aid. So if everybody has a reaction to a bee in the field, I know can save you and know how to use an EpiPen of various types. So I am well trained, ready to deal with any emergency now. Yeah, I work for Oregon Department of Fish and Wildlife. My field office is in Salem. I cover the Willamette Valley. We are participating in a multi-state grant with Rich's crew to do bee things, and super excited about the Franklin's Bumblebee in particular, although it's not really my area, but I used to work out of the Ashland area and have fond memories of working in that area. Although it's probably hotter than HE double toothpicks right now, but that's okay. And just really excited to be included in the project and working with great volunteers that I know are going out and looking at bees and doing good things. And if you're in the Willamette Valley, feel free to reach me out. I am at ODF&W, and I believe my contact details were given out by the ever efficient Rich. So thank you so much.
Thanks, Laura. Thanks for being here. We appreciate you too. Okay, we're going to jump in module 2. Leaf and I are going to split this one. He's going to start off by talking a little bit more in depth about the species distribution model that he developed for Franklin's Bumblebee. Let me see if I can share my screen here. There we go. Okay. Yeah, so I'm just going to turn it over to Leaf here. Leaf, let me see if I can give you control here as I did earlier. Okay, Leaf, you should have control of the slides. If you don't, just ask and I will advance them for you.
I think I've got it. There. All right. Good. Okay. So I'm going to present, as Rich said, about 5 slides here and then turn it back over to Rich. Some of these you've seen from the end of the last talk, but I'm going to give you a little bit more information about how we create those species distribution models. And I start with this slide that you've already seen to try to answer that question about range extent. So clearly, we know something, we have about 340, we have exactly 340 collecting events where one or more Bombus franklini was either observed or collected over the century and a quarter that of time that we've been paying attention. And so we know a lot about the range, but as I said, there could be extensions to the range if we find additional places where the bee occurs in Southern Oregon and adjacent Northern California. And so one of the ways that we can predict where to look, again, is these species distribution models. And as I said before, a species distribution model is a hypothesis about where a species occurs. It's based on presence localities for this particular type of species distribution model. So no absences, just records of the bee or other organism. And then those are the dependent variable. And then we bring all of this environmental and other sort of information in as predictor variables. So things that help to predict the ecological niche of a species, things that are related to temperature and precipitation, perhaps elevation, the aspect of the and slope of the ground and soil types and all of that good stuff, right? So we can take some educated guesses at where to go looking for these bees. We don't just have to look at the cloud of points and say that is where they occur. So again, here is that cloud of points. And again, the red ones are the only 2 or so observations that we would consider relatively recent, and the others are older. And as I described before, this is what that species distribution model looks like. Now, I'll say a little bit more in depth about the output here. This is a probabilistic modeling exercise. So it uses probability theory and Bayes theory, if you're mathy, to model the distribution based on the information that we give it. Each time you run the model, you get a slightly different answer because of that probabilistic nature of the model. It is picking a subset of the observations and modeling the distribution. Then it is using another subset to test the model to see how well it was constructed, how well it predicts. And so we average models, we average across multiple models to get a consensus, which is what you're looking at here. This is the mean of 5 models. We also use a model selection pipeline because we often start with many predictor variables. So so in this one, I think we have about 25 different predictors that we think could be of import to Franklin's bumblebee. 19 of those are called bioclimatic variables. Roughly half of them are related to weather station of observations of temperature and the other half of precipitation. And both of those sets are informed by remote sensing data from satellites that help us to fine tune, like land, the temperature of the land, of the Earth's surface in various places. So we crunch all of those those predictor variables together. And then we don't want a big messy model with a ton of things in it that don't have any predictive power. They actually reduce the statistical competence of the model. So we go through this pipeline to get to the smallest number of predictor variables that give essentially the best statistical fit. And I'll talk more about what those actual variables are in just a second. There we go. Oops, went too far. So after going through, I think, several different iterations of this model, starting with some 25 predictors, as I said, I arrived at this final model that just has 6 of them. And I'm just using codes for the names here, but I'll explain briefly what they are. The first 3, which I have named Bioclim 3, 5, and 6, are temperature related. So they have to do with the mean annual temperature in a particular place, with the comparison of temperatures at different times of the year, perhaps the temperature in the coldest quarter of the year or the warmest quarter of the year. So these are all different ways of slicing that weather station data, which is collected at points on the surface of the Earth and then gridded out, rasterized to a global coverage. And then the fourth one, number 13, is related to precipitation. The one called solar oct is about incidental solar radiation. And we have models of this for each month of the year. And I introduced, I think, 4 of those at each quarter of the year into this model. And it turned out that in the fall, the amount of sun that reaches the ground is important to predicting where this bee occurs. And then finally, this is a land use, land cover model, the USGS, bottom right-hand corner of those 6 predictors. The other thing I want to say about this slide is that it just shows in green the behavior of, or the values, it's a histogram or a density function of the values of those predictor variables in places where we already have Franklin's bumblebee observations, as compared to the gray, which is the quality of that variable at all of the locations in the area that we looked at. So if we draw a big circle around all those points, a few hundred kilometers in every direction, the gray represents the environmental characteristics of that whole area. The green represents just Franklinized habitats. And so you can see that the model found these variables are differentially important to predicting where to find Franklins, because it's somewhat specialized in one of these temperature variables, or in the case of land cover, it's associated with 2 of the 15 or 20 land cover classes, but not the other ones. If you are interested in that stuff, I can give you more detail, but that's a general idea of how we use these environmental variables to build these models. And with that, I'm going to turn this back over to Rich, and he's going to tell you more about what we are doing in this project with this model. So I think you're in charge now, Rich, if you want to go
Yeah,
ahead.
I am. Thank you very much, Leaf. Yeah, so what we have done is we've taken the species distribution model that Leaf developed, and we've put it on a map, and we've created a grid around that map, or around that species distribution model. And our goal here, right, this webinar is called Finding Franklin's Bumblebee. And so the goal here today is to organize a group of people, including agencies and including volunteers and nonprofits, to try to distribute survey effort throughout this species potential range to try to find it. We can't conserve this species unless we locate populations of it. And historically, Leaf alluded to this, survey effort for this species largely has been, dedicated along roadsides and in relatively accessible areas. But this whole Siskiyou Cascade ecosystem is large and diverse and wild. There's a lot of open land out there that potentially has never been surveyed. And so highlighting some of these remote areas and encouraging people to get out there is our goal today. And we're using a bunch of these tools to do this. And so I have taken Leaf's, as I mentioned, species distribution model. I've put it on a base map, and then I've created a grid, and I've laid that over the top of the species distribution model in hopes that we can somewhat follow the model of the Pacific Northwest Bumblebee Atlas project. And if you're not familiar with that project and you're just joining us sort of on a whim, that's a community science project that we have running in Oregon, Washington, Idaho. We've divided those states up into equal area grid cells, and we've encouraged volunteers to adopt those grid cells and then go out and follow a standardized survey protocol. Essentially, we've just carved out the grid cells in Oregon that overlap with the PNW Bumblebee Atlas, and we've extended it down through California to overlap with the potential range of Franklin's bumblebee. And if you look at this map, this grid that we've developed, you'll notice that we sort of extend the grid beyond the high probability areas of the species distribution model. And there are a number of reasons for that. Leaf alluded to the fact that
Hey,
this
Rich.
is a hypothesis. Yeah.
Sorry to interrupt, but I can't see your slides. I don't know if
Oh, well, that's good to know. You must have stopped sharing there, and I didn't notice it. Sorry. Will you let me know if you can see it?
Yes, looks good.
Okay. Very good. Thanks for letting me know that. Don't ever be sorry for interrupting me in a situation like that. Very good. Okay, so as I mentioned, we've had the species distribution model. We've overlaid this grid. We've extended the Pacific Northwest Bumblebee Atlas grid down into California, and the grid goes sort of outside of the species distribution model. And the reasons for that are that the species distribution model are a hypothesis. It's possible that, historically, survey effort for this species has been pretty small. We only have really a handful of records, 60 some odd records, I believe. So it's not a lot of information to go from. And so we may learn more about the species by surveying, beyond where it was known historically. We may find new areas. And also in this map here, the actual distribution of the species has been found sort of outside of the high probability area that Leaf mentioned. There are two records sort of down in this lower band of within California, FF 30 through 33. I believe there are historic records within that band, even though it doesn't show up as a high probability area for detecting the species in the species distribution model. So that's why this model looks like the way that it does, and why we've extended the grid sort of beyond what looks like the core of its range. So you can find this grid if you're following at home alone and have a couple of screens and want to look at two things at once. You're welcome to go to pnwbumblebeeatlas.org/ffbv. That's where you'll find this sort of focused effort for Franklin's bumblebee. I'm also going to walk you through that website in a minute or so, just to show you how to use those resources and how to use the map most effectively for planning and also for adopting maybe the grid cell of your choosing. So before I do that though, I want to roll through a couple of more slides. So once you've chosen a grid cell and you've decided you want to contribute to this project in one way or another, we're still at this question of where should we be looking for this species. And within these grids, we think it's a really good idea to survey largely on public lands. Certainly, if you own property and have private land, you're welcome to survey on that private land, and it's a great idea to survey on that private land. But you would need permission to survey no matter where you're going to be. So if you are going to survey on somebody else's, private property, you will need permission to do so, and you'll need that explicitly. So please follow all local regulations when you go out to do surveys, and making sure that you're respecting people's space and property and all posted signs and regulations. Within those public lands, the BLM and Forest Service lands are both great places to go conduct surveys. We recommend that you spend your survey effort largely in flowery meadows and riparian corridors. And within those flowery meadows and riparian corridors, we would suggest that you look on the key plants. And we'll talk about some of these key plants in a minute here. And Leaf has already mentioned some of them. But I also I think I'm going to stress this throughout the rest of this little module here, is that, just because, these places is where we found them in the past on flowery meadows and in riparian corridors, as well as on key plants, doesn't mean that you shouldn't be looking elsewhere, right? We need all eyes on deck here to find this species. And so we're going to have to think outside the box to a certain degree as well, and look in potentially what we may currently think as low probability areas as well. And we may very well detect it. And a perfect example of this is just a bit of a story from 2 weeks ago. I was traveling through northeastern Oregon looking for the western bumblebee, Bombus occidentalis. And I was out for 2 weeks. I did over 30 surveys in a bunch of different areas, mostly in flowery meadows and riparian corridors. Near the end of my trip, I was just northeast of John Day, Oregon, and I was wandering along a forested stream corridor. So I was in a closed canopy forest, and there were some sedges that were blooming right below me. It was the only thing in flower. There was no other flowers anywhere nearby. And sure enough, sitting on one of those sedges was a western bumblebee. And I was lucky enough to capture it and get a photo of it. But it would not have been a place that I would have conducted a survey for that species. And it was the only one that I detected in the entirety of my 2-week trip. So we definitely need to be thinking outside the boxes and potentially looking on resources that we may not associate with, as key plants for bumblebees. As Leaf mentioned throughout his talk, there are a lot of unknowns, and part of the reason that we're doing these projects is to learn more about them. So again, so within this now, how do I do this? How do I find flowery meadows? How do I find riparian corridors? How do I find public lands? Again, on the Pacific Northwest Bumblebee Atlas page /FFPV, I've put a layer in there that will allow you to look at the public lands and overlay that on top of the grid to help you try to find locations where you could go survey for this species. Likewise, you can change the base map of that map, and I'll show you how to do that as well, so that you can look at aerial photography and look for open spaces and open meadows. And then I'll also run through here in a minute after I show you the website. I will also walk you through sort of a pictorial of some of what we believe are the key plants for this species. So without further ado, I guess I've talked enough about this website. I should probably just pull it up here. So give me a minute and I will pull up the Franklin's Bumblebee web page and share it with you. Leaf, can you see this?
Yes, I can.
Perfect.
Good.
Okay, so here is the webpage. If you've been to the Pacific Northwest Bumblebee Atlas project before and you've adopted a grid cell or looked at our map, this should look very familiar. So you scroll down here and this is where you will see the map for finding Franklin's bumblebee. And you can interact with this map just as you would almost any other web map. You can zoom in and out using your scroll mouse or the buttons over here. And you can sort of navigate within this grid to look at different areas. If you want to look at aerial photography instead of a topographic based map, you can click on the different base maps here at the bottom, and then you can see aerial photography, which will allow you to see sort of open areas. And if you find that the species distribution model is obscuring what you want to see underneath there, you can actually turn the species distribution model off by clicking on the little hamburger logo here. And if you just click on the eyeball next to Finding Franklin's SDM, it will turn off the species distribution model, and then you can zoom in real close and start looking at what the landscape looks like under some of these polygons and under some of these lands. In addition to this, once you find an area, let's say I was interested in surveying this triangle right here, it's very likely that that triangle is not public land. It's very likely private land. But if you wanted to check this, you can click on this federal lands reference layer, and it will turn that layer on. And you can see, since nothing overlaid there, this is actually private land. But if I zoom out here, you can see that now I can see these yellow polygons here. These are all BLM lands within this. So it would be within these yellow polygons that would be great places to look. And then if you zoom a little bit further out, you'll see the light green polygons. And these light green polygons will be Forest Service lands. I'm going to zoom a little bit out for you here. You'll notice that the area around, well, you'll see if I turn this off, oops, if I turn this off, this is that area around Crater Lake National Park that Leaf suggested we were maybe a little suspicious of, but nevertheless, you'll see that the federal lands there is purple, and this indicates that this is National Park Service land, and it's not Forest Service land, and it's not BLM land. This public land here, while it belongs to us, national parks are managed a little bit differently than the BLM lands and Forest Service lands, and you would need a scientific collecting permit to do a survey within a national park. So please do not just head into a national park and decide that you're going to conduct a survey. You could certainly go into Crater National Park, Crater Lake National Park with your camera and take photos of bees on flowers. That would be a perfectly reasonable thing that you can do in a national park, but you cannot capture bumblebees for identification purposes on National Park Service lands. It's very possible that we could apply for a scientific collecting permit within this national park, but we've not yet done that. So please stay away from national parks at this point. If you're interested in applying for one there, we can help with that potentially, and certainly feel free to reach out to me if you need help with that. The same would be true on like Fish and Wildlife Service wildlife refuges and other areas like that. You may need explicit permission to do that. If you're ever in doubt about whether you need permission, you should reach out to the land manager and see what their answer is. They should be able to provide an answer for you. So that is basically how this map works. You can also turn off the grid cells and other things as well. You'll also notice here that one of these grid cells is a bit opaque, so which means I can't see 100% clearly through it. This means that somebody has already adopted this grid cell. And I say this above, I'll say it later in the presentation as well. We're part of the goal of this effort is to distribute distribute survey effort throughout the entire range of the species. But we don't want to let the fact that somebody else has adopted a grid cell serve as a deterrent for you if you really want to go do a survey in that location. This is just meant to serve as a guide. If you were looking to help in the best way possible, you might choose a grid cell that nobody else has adopted and try to go within that grid cell to find the species. But like I said, if you are interested in going to FF24 and you have your mind set on it, don't let the fact that somebody else has already adopted this grid cell deter you from going to do that. More eyes on the ground are better than fewer eyes on the ground. And just the fact that somebody has adopted a grid cell doesn't mean that they'll actually go conduct a survey there. So anyway, just let this serve as a guide and not as a deterrent would be what I would say about that. And then, so once you've gone through all this and you've sort of decided which grid cell is right for you, you can scroll down to the bottom of the page here, and this form here is where you would actually formally adopt a grid cell, which would then sort of put your name in our system as someone that's agreed to go do a survey for Franklin's Bumblebee. And it would turn that grid cell a little bit white up here, indicating that someone has already adopted it. So it would just sort of let the community of people that are interested in this project know that hopefully someone's going to go survey within that grid cell, so that potentially we can expend our resources in a way towards areas that are unlikely to get surveyed. And that's the purpose of all this. And then once you adopt a grid cell, you'll see a confirmation page. And there's some questions here at the bottom. One of these we're going to go over more in the next module, is just sort of acknowledging that this species, because it's proposed endangered by the Fish and Wildlife Service at this point, and it's a species of greatest conservation need in the state of California, there are state and federal protections that regulate how this species can be handled. And we'll go over that in more detail next. But it is important to recognize that, especially right now within the state of California, you will need to have your name on a permit to handle the species. And we're handling that. You're just going to have to jump through a few steps in order to get your name also on that permit. Once the species is officially listed, we will also probably enter a similar situation. But again, Xerces will take the lead on managing that permit, and you'll work with us to get your name on our permit. You won't have to get your own permit. And Jeff is going to go over this in more detail, and Hilary as well, in the next module. I'm just want to acknowledge that this piece is here before you officially adopt a grid cell, and that you recognize that this species does have sort of unique protections that other species don't at this point. And that's really it. This is the webpage that I've developed to direct survey effort. And then I'm going to jump back into my presentation. Feel free to explore that page on your own, either now or after the webinar. You can also just find it. I will just do one more thing here. In case you're not good at remembering URLs, you can also, if you just go to the pnwbumblebeeatlas.org, you can just go to get involved, and there's a Finding Franklin's link right here on the website. So you can find this page by just navigating normally from the normal web page as well. Okay, that's all I wanted to show you there. I am going to now go back to my presentation, which is really just going through a few more plants, or a few of these plants. Sorry, it's somewhat complicated to shift sharing, but I'm almost there. Okay. Leaf, could you just confirm you can see my presentation again?
Yes, I can. Looks good.
Thank you. Okay. So Leaf showed you this this slide earlier, which is the proportion of records that have been seen on different host plants. I'm just going to walk through these in turn. Each sort of, I put the common names over here in the same order that the genus is here. So we have California poppies. Coyote Mint is the common name for Monardella. Vicia is vetch. Centaria are knapweeds. Lupinus are lupins. Trifoliums are clovers. Eriogonums are buckwheats. Helleborus is hellebores. Ceanothus is California lilac or ceanothus. Melolotus is sweet clover, and Cirsium are thistles. And then I've put Nectar Robbing here as a question mark, and I'll talk more about that when we get there. So just briefly educating you as to what some of these photos look, or what some of these plants look like if you're not familiar with them. This is California poppy here on the left. The vast majority of records for Franklin's bumblebee have been found on this plant species. And then the second sort of most common is this plant, Monardella. There's a more eastern genus called Monarda that's called bee balm. This individual species here, or genus, is a little bit different, but very similar. And this is called Coyote Mint. And then we have vetch and knapweed, which are also two important resource plants for Franklin's bumblebee, at least where we have records. Although I just want to remind people that the vast majority of records for Franklin's bumblebee take place on roadsides. And if you've driven roadsides around Ashland, you certainly know that there's a lot of vetch there and a lot of knapweed. And so just because we're looking there and we found Franklin's bumblebee there doesn't mean that it's actually its favorite host plant. It may just be it was available where surveys were conducted. So both of these are weeds and non-native. That doesn't mean that we shouldn't be looking there, but we should just know that they're non-native. And then we have lupins and clover. Important that there are several different kinds of clover. Here I'm showing you red clover. There's also white clover, and there's also a host of native clovers as well that are certainly worth looking on. And then we have buckwheat and hellebores. Buckwheat is also quite a diverse genus, Eriogonum is, and there are many different types of buckwheat. This can also be a non-native plant, but there are some native versions of it as well. At least I believe there are down within the range of Franklin's bumblebee. Hellebores are a beautiful genus of plant. Very neat that we found Franklin's bumblebee on that genus. This is also a diverse genus, but these are often sort of hanging flowers. They can be brown to deep maroon, and sometimes actually don't even look like they may be blooming. So this is an important one to keep an eye out for if you see it in a landscape. And then we have California lilac and thistles. California lilac often will bloom in clear cuts down in that region. It can be quite abundant in a lot of different areas. And then this is a picture of a native thistle. At least it's native to southeastern Washington. There are also likely native thistles down, or I know there are native thistles down in the Cascade-Siskiyou area. There are also likely a fair number of non-native invasive thistles there as well, and both would be worth looking at. Leaf asked me to introduce this slide of nectar robbing, and I think this is a good idea. This whole subgenus of bumblebees, the Bombus sensu-strictu, are all short-tongued bumblebees, which means they have a relatively short tongue and can be difficult for them to access nectar resources within flowers that have a long corolla tube. This is a picture of the western bumblebee, Bombus occidentalis, nectar robbing a silene plant in northeastern Oregon. I came across this a few years ago, and this individual bee and several like it were, instead of visiting the flower from the front, how we would normally expect a bee to visit a flower, they were chewing a hole using their mandibles in the side of the corolla tube and then sticking their whole face down into that flower to drink nectar out of it. And this is called nectar robbing because they're not going to be helping this plant to reproduce, right? They're not going to be taking pollen from it, and they're not going to be delivering pollen when they return to it if they're just drinking nectar in this way. And the reason that I wanted to bring this up is that we have some potential observations of Franklin's bumblebee nectar robbing, but certainly it's very closely related taxa within North America are very well known for nectar robbing. So it's quite possible that Franklin's bumblebee may also do this. And so it's worth looking out on some of these plants like silene for an animal that may be nectar robbing. And so those are the likely plants that are worth looking at. Again, I know I've said this a number of times. I think it's an important point. It's just that what we found them on doesn't mean that's what we're going to find them on again. So don't restrict your searches and your surveys to these plants, but certainly if you find these plants within your search area, they are certainly worth paying attention to. They're probably all fairly attractive plants to all species of bumblebees, so they would likely catch your eye anyway. But please don't limit your search to these individual plants that we've talked about. You've seen this slide before as well. This is just to help guide us in terms of where, and this is the elevational gradient. You'll notice that the elevational gradient within Oregon is much greater than the elevational gradient within California. It's found, all the way down on the valley floors in Oregon, and we only found it at, relatively high elevations, close to 3,000 feet in California. So this is, again, just let this serve as a guide. Generally, it seems to be at higher elevations the further south that we go, which is a pattern that we might find a lot of bumblebees following, although this is such a small range that you do have to question whether we might find Franklin's bumblebee at lower elevations, or we may have historically had we looked harder within California. And then again, you've also seen this. This is just the timing effect of when we should be looking for Franklin's bumblebee. You'll notice there's a large spike in spring, and then a couple of spikes throughout the season. And the majority of records from this species come from Robbin Thorp, and Robbin was sort of well known for doing sort of monthly trips to this area to do his surveys in sort of May, June, July, and August. And I suspect that these spikes that we're seeing are not biological spikes, but they're probably just survey effort spikes. So I don't suspect it would sort of dip between that first high point and the second one that you see. And I know Leaf mentioned this as well, but I do think sort of the period between sort of June and July and August are all likely the best times to look for this species. And certainly at the lower elevations in and around Ashland and further north from there, you could focus earlier in May. Might also be a good time to look for this species, sort of the more north that we get. And then again, I'm going to say it one more time. I've probably said it too many times, but just remember that what was isn't necessarily what is. We haven't seen this species in 15 years. There have been a lot of fires in that region. Climate change is altering when plants bloom and how plants bloom. Other species have been moving to higher elevations, or at least there's some basic evidence for that. So don't let this, if your elevation or where you're going to is slightly above or below what you see on this chart, or you're looking a little later or in one of those dips, don't let that deter you. It doesn't mean that we're not going to find this species there. This is just sort of meant to serve as, we're just giving you all the information that we have, but it is not comprehensive, and you may contribute to our knowledge of this species. So anyway, it's worth searching outside of the windows that you see here. And with that, I am going to end this module. And we're going to move on to talking about survey and identification. But before we do that, I will quickly check in here and see if we have any questions that need to be addressed.
So Rich, one question that someone asked early on in this module was if you could just explain the heat map colors in the slides that we've used and on the website. And I answered the question in text, but I thought for everybody's benefit, you could just explain that. And,
yeah,
and it's also visible on the website, yeah.
Yeah, you can look at the, another option I didn't show you on the map on the website is to look at the legend, and it does indicate that. And I'm sorry, I probably should have been more careful and used the same color scheme that Leaf did, or the same heat map that Leaf did, or vice versa. We probably should have used the same one. On the map on the website, the cooler colors represent a lower probability or lower suitability of that habitat type. And the warmer colors with yellow sort of being hotter than red indicate a higher suitability of that habitat area. So But again, take that for what it's worth. If you look at that dot over Crater Lake, a lot of that high proportion area is actually over open water. And I'm pretty sure we're not going to find the species in open water.
Yeah. Okay, here's another good question. Valerie asks, is there any evidence of photoperiod inducing any kind of life history tactics? Has there been post-fire monitoring of known sites? Climate change and impacts to phenology of host plants for foraging. What is the outlook for the species? So if you could talk a little about phenology and photoperiod as well as fire and climate change.
Yeah, that's a big question with a lot in there. A good question. I don't know very much about photoperiod influencing behavior in bumblebees. From what I understand, it's more sort of growing degree day or heat related. In terms of photoperiod actually indicating there may be some indication for sort of senescing at some point. There may be some cues, but I'm not aware of anything explicit about that, Leaf. See, you've turned your camera on, which probably means that you are. So I will let you go in here.
Well, I have one thing to say about this. So first of all, no, I'm not aware of, unfortunately, Valerie, of photoperiod specifically and in the narrow sense influencing bumblebee behavior or life history. But in a more general sense, yes, bumblebees do monitor, do keep track of time in various ways, right? And one of the really interesting and potentially worrying aspects of climate change with respect to bumblebees and plants, bees and plants, is that both plants and their pollinators go through some sort of diapause period in the colder or drier parts of the year, and then they have to figure out when to bloom, when to grow, and when to emerge from the ground. And so bees can track some of the same cues as plants, namely temperature. So if a bumblebee is underground in its diapausing chamber, it's sensing the warming of the earth in springtime, just as the plants are. But plants also have access to solar cues in terms of what season is. And so what I'm getting to is that climate change is making spring happen earlier, and the cues, the set of cues that bees use and that plants use to develop and grow are different, or they're overlapping, but they're distinct. And so there's been a lot of research on this potential phenological mismatch, as we call it. And thankfully, many of the papers so far on this conclude that there is no mismatch yet, that bees are emerging around the same time plants are flowering, even though both are emerging or flowering much earlier than they used to. However, there are some studies that show a mismatch that has resulted in lower plant reproduction and potentially lower resource acquisition by bees. So phenology really matters, and bees and plants' ability to track it is really critical. It's one of the things that we worry about with climate change.
Yeah, I would say the other major threat with climate change is just as snowpacks melt out earlier, some of our high elevation wetter meadows that are dependent on snow melt to stay wet are drying out earlier, which is shortening the length of time at which flowers are blooming within a meadow. And I don't know if we've talked about this, but in his ecology section, I may have missed it, but it's about 5 weeks from egg to adult, 4 to 5 weeks. And so if you think that sort of, a queen needs a worker caste, and then also beyond that needs a second caste to start developing reproductive members of the colony, the queens and gynns, we're talking about probably a 10 to 12 week period that a bee needs flowering resources. And so as that season gets shorter, particularly at higher elevations, if things are melting out earlier and drying out faster, the seasons may get too short to support bumblebee populations. And this is a selection factor. It doesn't mean that bumblebees are doomed. It could be that some species or individuals within a species could adapt to have a shorter developmental period or something, depending on how fast these changes happen. But this would be another thing that we would be concerned about with climate change and bumblebees, is this shortening of the season, and whether there'd be enough resources even after the new gynns are produced, whether they can have enough food to build up the fat reserves they need to make it through the winter time.
Yeah, Rich, two other practical questions from two folks about the website and the map. One person is wondering, could we add a roads layer that folks could overlay on the species distribution model and other information? And then also, is there an elevation or topo layer available on the website? And so I can say there is a topographic map there, but we don't have a digital elevation model. But Rich, do you want to talk about whether you think those would be good ideas to add?
Yeah, well, if you can zoom into the map in the topo version, and it'll have topographic lines that have the associated elevation and feet on there. So you should be able to see that there. In terms of a road map, that topo map does have roads on it, but it's not sort of a Google travel oriented map. I can make a Google My Maps version of this, as I have the Pacific Northwest Bumblebee Atlas, and that would provide the option for you to export this grid as a KML file if you were interested in doing that. I see another question there related to that. And then you could also use Google's different map types for travel planning as well on that page. But I can only, in this web app that I've developed, I can only put 2 different base maps available for you. And I think I chose the 2 most appropriate ones, but I'm happy to make that Google My Map available. It's not available right now, but I can do so fairly quickly by the end of the day tomorrow.
Good. Do you want to take one more question, or shall we move on?
We should probably move on in the interest of time. I just looked, we're about 10 after 3, and we still have quite a bit of material to cover. So yeah, maybe we'll try to type the answer there, at least for that next question.
Do that.
Great. All right, so the next thing we're going to move on to here, folks, there's a lot here. You're asking a lot of great questions. I had planned on trying to putting a break in here somewhere, but I don't think that's going to happen. So you're just going to have to bear with us. Feel free to go take a break if you need to, although this next section here is going to be, I'm going to ask Jeff Everett to jump in and present next. He's going to be talking about sort of the regulatory side of the Fish and Wildlife Service, what their goals and hopes are. And I would encourage you to stick around for this part. I think this is really important. One of the key pieces, I think, to this webinar is hearing from the Fish and Wildlife Service about their hopes and dreams for a project like this. And then Hilary Sardinius, who works for the California Department of Fish and Wildlife, who introduced herself earlier, is going to talk about the regulatory side briefly from California as well. So I would encourage you to stick around for the next 15 minutes or so and just hear this side of things. And again, Jeff, welcome and thanks for being here. I'm going to turn it over to you.
All right. Going to share my screen. Bear with me just for a moment. All right, can you see my presentation okay? Yep.
You're good.
Okay. I'm going to breeze through this pretty quickly in the interest of time. And some of this is going to be a little bit duplicative with some of the information you've already seen. And then as we've listened over the course of the last couple of hours, there's a couple of things I'm going to add just as anecdotal discussion points that aren't necessarily captured in my slides. So we're going to dive right in. I'm going to talk very briefly about bumblebees and the Endangered Species Act, and I will probably skim very briefly over the species status assessments, as it's not necessarily totally germane to what we're trying to accomplish today, but interesting anecdotal and background information. And then talk about some of the other work we're doing, trying to support this larger effort of finding Franklin's bumblebee, the where, why, and how of where we're searching for them, and then hopefully roll into questions a little later on. And so just briefly with the ESA and bumblebees in general, there's only 6 species of bumblebee that have been petitioned, although I've heard rumors recently of a 7th, over the course of the ESA. And so since 1973, when the ESA has been around, there's been over 1,600 species across a wide variety of taxa that have been listed in the United States. And there's only been one of those that's a bumblebee, and that's the rusty patch we talked about earlier. Franklin's is likely at this point to be the second bumblebee added. And then there's a couple that are navigating their way through the ESA process, everything from being originally petitioned to having this SSA document, and then moving from a final delisting decision into recovery. And so the Fish and Wildlife Service largely manages this with this whole process with a wide variety of conservation partners, experts, stakeholders, landowners, both federal, state, and private. And there's a lot that goes into any one of these specific decisions. And so, like I was saying, Rusty Patch is listed. Franklin's is moving closer and closer as time goes on to finally being listed. And that may have some ramifications for our work we can talk about a little bit later. And then there's a number of others that are navigating the process as we speak now. And so point being, the application of the conservation provisions of the ESA to bumblebees is relatively new. It's also further complicated by the fact that there's a lot about these critters that we just don't know. And I think that's been really well portrayed over the first part of these presentations. There's critical pieces of their life history and how they do what they do that we don't understand as well as we would like to in order to inform meaningful conservation delivery, especially for something that is relatively small, relatively cryptic, is constantly on the move, and have very big ranges, Franklin's notwithstanding. To give you an idea of some of the widespread declines we're seeing in bumblebees in general, another closely related species to Franklin's bumblebee is the western bumblebee. And the probability of occupancy across the whole range of the species. And so this is the northern subspecies, which goes all the way from the Aleutians. And then there's a little zone of overlap in Western Canada and then down into the Western lower 48. These are primarily the WAFWA states here, the Western Association of Fish and Wildlife Agency states. For such an incredibly wide-ranging species to drop in overall predicted occupancy at such a profound rate over a relatively short amount of time is really alarming. And this is what we're seeing with a lot of our other species. Obviously, we haven't seen Franklin's very closely genetically related to Westerns in the last 15 years, but we're seeing patterns of decline across very similar species throughout many parts of North America. I'm just going to skip this altogether because this, this slide, I made it and it still gives me a headache. It's a kind of an illustration of the convoluted process between petition and final listing, and then talks a little bit about the SSA. And the take-home point here, leading to the SSAs, this is a similar process, but the SSA is the species status assessment. It's that biological document upon which we make some of our ultimate decisions regarding a species status. The SSA is a living document. They're added to and developed over time. But importantly, the SSA is not a decision document. So there's a lot of work that goes into these. They continue to get things added to them as we learn more or find more species. And it's a process that is constantly evolving. The take-home point from here is that since the Fish and Wildlife Service adopted the SSA process a number of years ago, I think 2017, right in there someplace, there's been about 200 SSAs that have been written across a variety of taxa, and only 3 of those have been written for bumblebees. So similar to the provisions of the ESA, bumblebees are relatively new to the SSA process. And when using this model to help inform species status decisions, those uncertainties, the gaps of information, the things we really just don't understand well about the critters really comes through. And so there's a lot that goes into these, and a lot of unanswered questions remain. We've gone over this for the most part. So I'm going to breeze through this and get to my next slide. Again, Dr. Thorpe, and we've seen bits and pieces of this graph. This one takes it back from the historic high in 1998, I believe, and there were when this really started kind of dropping off, and then the last known observation in 2007. The point here, however, is that they've never been, Franklin's in particular, has never been very abundant on the landscape. These have always been relatively rare as compared to some of the more abundant species, and somewhat difficult to find. And so one of the points I'd like everybody to take away here is reinforcing something Rich was describing earlier. It's don't go necessarily with California poppy in this elevation range. These things could be just about anywhere within the historic range of the species. We've got a lot of ground to cover. And one of the most important things, if not the most important thing we can do for Franklins right now, is find them on the landscape. So there's a lot of things that influence the species, and disease and pesticides certainly being two of the big things that have been documented as having impacts to many of our North American pollinators, bumblebees in particular, but a wide variety of other things. We've talked a little bit today about small population dynamics, regulation, like the import and export of other bees for commercial pollination services, is potentially a pathway for introducing disease. Competition from non-native bees is an issue, and then a wide variety of habitat factors are playing out in real time across the range of most of these species that we're interested in. And one thing I want to point out is that it's not any one particular thing that is going to solve the larger conservation issue for these critters. It's a combination of these factors acting synergistically. It's kind of a death by a thousand cuts kind of thing. It's not like if we just ban pesticides, the bumblebees are going to become, much more abundant on the landscape suddenly. There's a lot of conservation work to do, and a lot of ways that even just little bits and pieces of conservation delivery in all of these areas can result in meaningful gains for the species. The current condition of Franklin's bumblebee, like we've said, we haven't seen it since 2006. And so we can't at this point identify existing known populations on the landscape. And because we don't have any on the landscape that we know of, it's very hard for us to predict what things are going to be like in the future. So we conclude our current SSA for the species by saying that there's no detectable level of viability for the species into the future, which leads to a huge question: is Franklin's extinct? That's a very difficult question to answer. There's a lot of case history when it comes to species that have been declared extinct and then rediscovered in the wild. It actually happens all the time. It recently happened not that long ago. There was something in the media about an elephant shrew that was rediscovered recently in Africa someplace. I'd have to dig through the media to find it. But this kind of stuff happens all the time. And there's an average of over 60 years between when something is last seen on the landscape and when the Fish and Wildlife Service, for a variety of reasons, says it's actually extinct. And we're only 15 years into Franklin's. I'm going to come back to that point here in a minute. This information I'm presenting is slightly dated. This is from some similar work that was done a couple of years ago, but simply not seeing it for a relatively short amount of time, again, when we're looking for something that's quite small and moves around the landscape and was never terribly abundant on the landscape in the first place, believe it's way too early to just write Franklin's off as extinct. A good case in point for this, and Rich had Leaf had a picture of Fender's in one of his early presentations as well, is Fender's blue butterfly. First described in 31, last seen in 37 and discovered not that far from here, 52 years later. And it was then listed. The Fish and Wildlife Service, along with a wide variety of conservation partners, delivered a conservation program that was quite extensive for this species. And we just recently proposed to downlist this from endangered to threatened because we've achieved some milestone conservation recovery objectives and goals, some really outstanding work. And this is the kind of thing we would love to do for Franklin's. But again, the first thing we need to do is find them on the landscape. And so the Fish and Wildlife Service has a kind of a parallel habitat modeling and mapping program to what Rich and Leaf were presenting earlier. And this goes to some work that some of our directors, fellows, interns did last year. We had planned to get them in the field and looking at all these high priority areas, and then COVID threw a wrench into everybody's plans for last field season. So our DFPs spent the summer digitizing based off of aerial photographs, all of the potentially suitable Franklin's bumblebee habitat within the historic range of the species, using a couple of different GIS layers, but trying to key out those areas where we know we found them in the past, those kind of higher elevation and therefore publicly owned to a large extent. Alpine meadows and places where we have snowmelt catch basins and kind of open timber or open alpine meadows with a wide variety of floral resources. And so our DFPs did all this work last year and came up, again, looking almost exclusively at the federal land. There's about 180,000 acres. This is, again, a very coarse first look at this of habitat that needs to be surveyed for Franklin's. I'm going to show a little bit more about this here in the next couple of slides, but before I do, I want to say that this is a relatively recent product, and we're in the process of refining it in real time this field season. And the next step is to match this up with the habitat distribution model that you saw earlier in the webinar today, and really kind of narrow in some high priority areas to focus. That work is a little ways down the road, and to Rich and Leaf's point earlier, we still need to look everywhere within the historic range of the species and cover that, cover as much ground as possible, because as I hope everybody understands now, they're likely to pop up in places where we're not necessarily keying in on the most important habitat factors, like looking only at a certain elevation range on California poppy, for example. Most of the mapped habitat is in Jackson and Siskiyou counties. And based on our work talking to the Forest Service and the BLM, since they're the primary federal landowners here, we've looked at roughly 5% of this from a standardized survey or a bumblebee or even Franklin's bumblebee specific point of view. So we've got a lot of ground to cover out there. This is mostly old news now, but based on where the historic range of the species, the bulk of the big habitat is in California in these two counties, and then Jackson and Josephine. Klamath, there isn't quite as much. This is some data that's been refined a little bit from the earlier model, but you can see that the bulk of the good habitat lies quite a bit of ways above the valley floors. And so we're looking at some of the higher elevation and federally owned land here. And then total suitable acres by habitat by county. Again, Jackson jumps right out in California, and then Siskiyou and Trinity in California. So I'm sorry, Jackson and Oregon. So there's a lot of interesting information here that'll help dive us into where we should be looking. And that's led us to the second iteration of this. All those polygons have been refined to include a wide variety of priority factors, including proximity to historic Franklin's locations, reasonable amount of access based on existing road and trail networks, and a couple of other factors. And so this is where our very hardworking DFPs and a lot of the federal agency folks are focusing their work this year, is looking at some of these higher priority polygons. And this isn't quite ready for prime time yet. We're editing this and making some ground truthing adjustments to the polygons based on the field work this season. But the goal for the end of the season is to mash all this data together with the other mapping efforts and really target some high priority areas. Ultimately this year, we're also, all the survey data that we collect will feed directly into the Pacific Northwest Bumblebee Atlas, as well as the Oregon Bee Atlas, which is bigger than bumblebees. But all the survey work that we're doing uses the standardized survey protocol and is all digitally linked up via a couple of different applications. And so at the end of this field season, we'll have the ability to share the information that we collect with some of these other larger citizen science efforts. And it's a demonstration of the strength of the partnership and just reinforcing that we need as many people looking for these critters in as many places as possible. This year is by far the biggest year in terms of interest and participation, people seeking out places to go find Franklin's bumblebees and Western bumblebees and our other target species. And more people going through the training programs, attending webinars like this and those sorts of things. I also wanted to point out that in addition to the surveys, we're piloting some other work with the USGS to use eDNA methods to possibly detect our very rare bees on the landscape without actually having them in hand. And so that technology is in its infancy. We're refining some of that this year. Hopefully we get some positive results out of it, and we might be able to find some of these critters on the landscape without actually even identifying them, proving them that they're out there using genetic markers. So that's also in its infancy, not quite ready for mass application across the historic range of the species. But if we can make it work on a small scale, we'll look forward to rolling it out on a much bigger scale as well. So with that, I think that's my last slide. I'm going to stop presenting and talk just very briefly about the regulatory environment with Franklin's. And so Franklin's was proposed to be listed as endangered in August of 2019, and that kicked off a 12-month process. So we were supposed to have Franklin's final listing determination made in August of 2020. August of 2020 was a very interesting time in this country in advance of a contentious national election. And so some things kind of got stalled in process, for lack of a better way to describe it. And Franklin's, the final decision for that is one of those things, along with a whole host of others. And so there's recently been litigation filed against the Fish and Wildlife Service to move some of those packages that have languished since the transition along as we are regulatorily required to do. And so the Franklin's package is slowly moving through its final stages of approval at the headquarters level, and eventually will be published in the Federal Register. Headquarters still hasn't given us what the final version of that looks like. I believe it will look awfully similar to what we originally proposed, with some changes potentially associated with the comments we got during the public comment period. But the point being is, even if it published in the Federal Register tomorrow that Franklin's bumblebee was going to be finally listed, it would have an effective date of 30 days at least after the publication date. And so if it happened tomorrow, we'd be in the middle of August before Franklin's would be officially listed, most likely later. And so to the point about needing permits to handle it or to capture it, technically speaking, yes, that would legally be required. But I'll be working very closely as the species lead to do the consultation work with Leaf and Rich and the rest of the gang, so that citizen scientists and the folks doing this incredibly important survey work, again, the most important thing we can do for them right now is find them on the landscape, would have the protections that they need to do the survey work. So I don't want to have anybody worry about what they're doing out there causing a problem, or being in a situation where we have a really delicate take issue. I think we can navigate the permitting process there. One word of caution, however, if somebody finds Franklin's bumblebee out there, I would encourage everybody to photo it, handle it extremely carefully, photo it as much as we can. And then from there, we would take the photos, run them through taxonomic experts for verification. There's a number of taxonomic experts we have at our disposal to do that, several of whom are with us today. And then we would proceed from there. We would not collect a specimen from the wild at this point. If we do find populations on the landscape, there's the possibility of doing some voucher specimen collection in future years once we know where to look. That could update the genetics and the taxonomy, but we're not in a position to do that this year. So if you find it, photograph the heck out of it, send them to us, and we'll go from there. And with that, Rich, is there anything else you'd like me to cover?
I don't think so, Jeff. I think that was great. And I appreciate you giving that perspective and sharing your opinions and thoughts. I guess they're not really opinions, but sharing the status with us. I don't see any questions that are directly related to that, Jeff, so I'm gonna pass it to Hilary and let her talk about, briefly talk about things from CDFW perspective. And Hilary,
Sorry, for some reason I can't click present slideshow. It's very fun. Let's just do all of screen too, and then maybe I'll be able to hit it. Nope. Interesting. Even when we have tests, it never works the same.
hey, Jeff, while she's working through that, someone's asking for your email address in the Q&A. Is it okay if I post it there?
Hilary, we can see your file, but it isn't in presentation mode, slideshow mode yet.
You're muted now too, Hilary. We can't hear you if you're trying to speak.
The bar at the top is covering the thing where I can do the, I don't know why. There we go, slideshow.
I had a similar challenge with the board. There you go.
Fun. I don't know. I've never had that happen before. All right. Hey, everybody. Thanks for having me. If you want to do sampling of franklins in California, which we hope you do, then you will need a scientific collection permit. And so I'm just going to talk really briefly about that. And then hopefully you can just jump right in. So in California, we have a list of terrestrial invertebrates of conservation priority, and franklins is on that list because of how rare they are right now. And so you would need a scientific collection permit. However, Xerces, as I already mentioned, has applied for one, and that will be finalized next week. And in our first case ever, we are allowing community scientists, so folks like you, to be added to that permit. So that is pretty great, and it should be pretty easy for you guys to do that. And I also just want to note that we are working with the Fish and Wildlife Service, so with Jeff, to make sure that our permit conditions are really similar. And so we are not allowing, again, any vouchering. We want you guys to follow the protocol that Xerces is about to go over. I also just want to note, like, generally there are numerous other bumblebee species that are on our list of priority species, other bee species, other pollinators, even non-pollinators. So I just want to let you know that you should not be going out and sampling anything that's on that list unless you have a permit. And if you are interested in getting a permit, here is our website. And as soon as I'm done talking, I will pop that into the chat. You can also contact Justin Garcia, who is our scientific collection permit lead, if you have any specific questions. Okay, so if you want to go under the Xerces permit, what do you need to do? Well, first and foremost, you need to get training in how to ID franklins and also in bumblebees. So that is what this webinar will be serving as. You'll also need to pass a quiz that is related to the survey protocol, as well as a scientific collection permit conditions. And Rich, I believe, will be offering that quiz at the end of the webinar, but it should also be on that grid cell adoption page, along with this PDF version of my very awesome no picture agency presentation. So if you need a refresher on what you need to do, it will be there.
Worried we may have lost Hilary. Hilary, are you with us?
Looks like we lost her.
Did I just get disconnected?
Hi there.
Well,
Yes,
we
you
can
did.
see you now, but your screen stopped sharing. I wonder if maybe your bandwidth of showing your video and sharing the screen at the same time was too much. You might try turning your video off while you present. It might help.
There you go. Can you guys see my presentation again?
Yes, we can.
Excellent. Sorry about that. Okay. So I'm not sure where I stopped, but I just wanted to say you need to have your permit with you at all times. When you're in the field, you can have a digital version, you can have a print version. This is in case you get stopped by law enforcement, including a game warden, be able to say, hey, I'm doing this legally. And again, as a reminder, we don't want any killing or harming of francolins, so please follow the protocol very closely. And if you do happen to find them, which we would be really excited about, then you need to report that to us within 3 days. You can also report that to Xerces, and they can report it to us since they are the lead on the permit. There's a few other things that are in the scientific collection permit that you would need to do. Somebody had asked in the question and answer, how do we prevent disease? Well, one thing you can do is you can clean your nets or your vials in between when you go to different sampling locations so that you're not transmitting diseases from one place to another. Something that we also recommend is just to spray your shoes with ethanol. That isn't necessarily for bumblebees, but there's rabbit hemorrhagic disease, sudden oak death, right? So that's just good protocol. If you happen to find a Franklin's bumblebee that is already dead, don't know how likely it is, but you are allowed to salvage that. So you would want to photograph it to record the condition, email Xerces and us for more direction, place that specimen in a freezer, and we'll tell you how to proceed with getting it to us so that we can figure out where the specimen should go. You'd want to collect GPS coordinates, the date and time that you encountered it, details on the condition of this specimen, what you think maybe was the cause of death, if that's apparent, and any other observations. And then again, we really want you to be as cautious and careful as possible when you are handling the species, photographing it, etc. But if it does happen to accidentally die for some reason, then you need to report it to us immediately. And then you need to cease all survey activities until you get more guidance from us. I just, Jeff talked about Franklin's being listed soon under the ESA, and the question came up earlier, will bees be listed under CISA here in California, right, the California Endangered Species Act? And I just wanted to say that, so those 4 bee species were petitioned to be listed. We started our species status review. That was halted when we were sued by the Almond Alliance and some other folks. Basically, they were saying that they didn't think that the Fish and Game Commission had the authority to list terrestrial invertebrates because the definition of invertebrate in the code is under fish, and so only aquatic invertebrates were what were thought to be what we had authority over. A judge concurred with the folks suing us last November, and we have appealed, and so we're currently awaiting that decision, and we'll know, whether we'll keep it going or if there will need to be a legislative fix or if we just don't have that authority. So with that, I'm just going to wrap up real quick. I really do hope that you guys participate in the effort and that getting on the Xerces permit does not create a barrier to entry. We really do want folks to find it. And we think that this process that we've come up with of just folding you into what they're doing will make it a lot easier than it would be if everybody had to go out and get their own. The last thing I do want to say is, like Rich said, when you're out there sampling, make sure that you have permission to be on the properties. For example, if you wanted to sample on CDFW wildlife areas, then you would need to get a letter of access. So just make sure that you're allowed to be doing it. And here is my contact info if you have any questions, but it will also be in the permit a bunch of times if you're here in California. So thanks so much. Appreciate you having me. I think you're muted, Rich.
My computer thinks I'm muted too. I just got a notification that told me that. I think I need a permanent notification for that, a permanent notification for that. Okay, so this is where we are. It is, it's 3:40, which means I have 20 minutes to talk to you about survey and ID, which is not sufficient time. And I am aware of this. I suppose I could do one of a couple of things. One is I could end on time and just share with you a bunch of links, but I'm not going to do that. I'm going to keep talking today. And the reason that I'm going to keep talking today is that we're recording this, and this is kind of our one chance to record this webinar. And so I may go as much as 10 minutes over, potentially even a little bit more than that. I recognize that this may not be doing what I told you I was going to do when you signed up for this workshop. And if you're only available until 4 o'clock, I apologize that you're going to miss the end of this presentation. But I feel like it's the best thing to do is to get this recorded so that other people that want to see this material in the future can see the entirety of it. I am also going to share, I'm going to cut out some of what I was going to talk to you, and I'm just going to share a link with you about that. So here in the chat, I'm going to put a link to all of the training modules that are available in the on the Pacific Northwest Bumblebee Atlas page. I just shared a link there that will take you to a page that is full of training videos, everything from Bumblebee ecology and conservation to very detailed sort of Pacific Northwest, species by species, how to identify them and what features that we look at. And you can go there and view that material in more detail than I'm going to be able to cover today. So anyway, I just wanted to let you know that I intend to go over today. I recognize that this may be not quite respecting your time, but I do think in the interest of everybody that this is the best way to proceed. If you are interested in surveying in California, you now know that you will need to be listed on Xerces permit, which will be active next week, which means you'll need to take a quiz. I will be delivering that quiz at the end of this presentation. If you have to leave before that, before the end of the quiz or before the end of the presentation, because you needed to leave at 4, I completely understand and appreciate that. Please send me an email. My email address is going into the chat right now. And I will email you the quiz, and you can take the quiz at your own time after this webinar. And again, I'll just apologize that I'm going to have to go over. Without further ado, I think I'm just going to jump right in here and start presenting. Leaf, can you see my title slide here or somebody? Hello. Can someone let me know they can see my title slide, please?
Yes.
Okay.
See it.
Thank you. All right. Before we go further, I just want to just take the time to explicitly say that this webinar is possible because of a grant from the Oregon Department of Fish and Wildlife that's called Oregon is Alive. It is the Oregon Conservation and Recreation Fund. It's a fairly new program. The URL is oreganisalive.org. and it is their contribution to the Xerces Society from a grant we got that makes this webinar possible. So I just wanted to explicitly acknowledge that they made this possible, or that this partnership made this possible, and that this is also funded from a wildlife sport and fish restoration, or sorry, the wildlife and sport fish restoration program from the U.S. Fish and Wildlife Service, which is a competitive state wildlife grant program. And just want to acknowledge those funders in addition to the partners that have introduced themselves to you today. Okay, I'm gonna jump right in here to identifying and photographing bumblebees, and then we'll talk a little bit also about how to survey them. I had originally intended to share background information about how to sort of distinguish a bumblebee from the other group of insects that you might find visiting flowers. I've decided to skip that in the interest of time. But again, at that URL that I shared earlier with the training modules, you can find that material. It's already recorded. So please, if you need that refresher or you need that information about how to distinguish a bumblebee, please go view the appropriate modules on that page that I shared with you. So as I go through this, I'm going to talk about different parts of the body. We'll be talking about the head, the thorax, and the abdomen. Bumblebees, like all other insects, have these three body parts. The head is pretty clear. The thorax is where the wings and the legs are attached, and the abdomen is behind that. The abdomen is broken up into tergal segments. Those are the T's that you see there, T1 through T6 in females, and males actually have an additional abdominal segment. So they have 7 abdominal segments. There are also corresponding sternal segments on the underside of the abdomen, but I should not be talking about those today. I'll just be talking about the tergal segments. The other major body part that we will talk about as it relates to Franklin's bumblebee is cheek length. So we look at the space below the eye and above the mandibular hinges. And you can see an example of this on this web page here, or I'm sorry, not on this web page, on this page here, where on the left you have a species with what we consider a long cheek. And on the right, we have a species that we consider a short cheek. Let me just turn on the laser pointer here. So here you can see that below the eye, the distance between the eye and the mandibular hinges is much longer than the width of the mandibular hinges themselves. Whereas here, the width of the mandibular hinges is actually a longer line than the length between the eye and the bottom, and that's that space below the eye and the mandibular hinges. So this is the case with Franklin's bumblebee. This is actually not Franklin's bumblebee over here. This is the Western bumblebee, which is again its sister taxa, but has a similarly short cheek. So when you are taking photos or looking to identify Franklin's bumblebee, this is a photo that will be very helpful if you take a photo that allows us to see this space. Or if you hear me talk about sort of a long cheek versus a short cheek as I go through some of the next slides, this is what I'm talking about. There are some fantastic identification resources that are available for the Western United States that wouldn't have been if we'd been doing this a decade ago. John Koch, Jamie Strange, and Paul Williams put together this book on the left here called Bumblebees of the Western United States. The Forest Service, I believe, still has the PDF of this available from their website. You can download it and just print out the pages that you need from it. It's also a great reference guide. There's a key to females in the back of it. The one thing that is not bad about this book, but potentially not great, is that the taxonomy is a little bit out of date, or at least it's different from the book on the right. The book on the right is sort of the authoritative guide to bumblebees for North America, but it includes a lot more species. So you're going to have Eastern species in there as well, and it's a little bit more to filter through. But it has the most recent taxonomy and is up to date with updated distribution information and more conservation information, et cetera, et cetera. There's also a dichotomous key in the back to both males and females. So it's sort of the authoritative guide, but potentially the one on the left, the Western United States might be a little bit better of a field reference for you as you go through this. As part of the Pacific Northwest Bumblebee Atlas project, I've also developed this pictorial guide to, or this cartoon guide really to bumblebees. And it's important for me to share with you that this is a overly simplified vision of what it's like to identify bumblebees. While this sort of simplifies it and suggests that every bumblebee can be identified by their color patterns, this is not true. There is a lot of intra-species variation. So lots of species have different color patterns that aren't necessarily represented in this guide. And an example to that here, just even on this guide, you'll see that Bombus rufocinctus here is both found in the section with red on it, and it's also found in this section with no red on it. So different species have a lot of variation, even beyond what you just see here with Bombus rufocinctus. I believe there are something like 26 different color patterns in the guidebooks I just shared with you for this species. So let this cartoon guide that I've developed serve as a reference guide for you, but it is not authoritative. It is really meant as an entry point for you to take the species that you're seeing out in the wild and maybe come up with a name so that you can go to those more authoritative reference guides out there. This page that I'm showing right now is available on our website at the Pacific Northwest Bumblebee Atlas under resources. You should be able to find the pictorial ID guide. I'm not going to go through all the species today. I'm just going to go through basically how to identify Bombus franklini and the photos that we would like you to be taking. And then I'm going to talk about survey protocol. So Bombus franklini is, from what we know about it at least, has a fairly consistent color pattern. There's not a lot of variation that we know for this species. And some of the things that we're looking for are just a tiny hint of white back here on the 5th tergal segment. So remember that the abdomen is broken up into 5 different segments. And here on T5, we're going to find hints of white or pale hairs. And those white or pale hairs are, from what we understand, divided down the middle by some black. Sometimes it's an incomplete break, and sometimes it's a complete break. But mostly what we're looking for are those pale white hairs on the T5 of, or the 5th tergal segment. In addition to that, and actually the cartoon that's drawn here doesn't display this properly, is their thorax has a horseshoe appearance to it. So in this picture guide here, which actually comes from the bumblebees of the Western United States, this hole should probably be cut out of this cartoon guide and should be black. It's this very unique horseshoe pattern that I have not seen on another North American species of bumblebee. It's very distinct. I will show you more pictures of it later, but these are the 2 features that we're really going to look for this species, is the horseshoe on the thorax and pale hairs only on the 5th tergal segment. What photos, if you do find the species, what photos should you take? It should be quite clear that detecting this species is the most important thing to do. So whatever photos you can get are perfect. If you are lucky enough to actually capture a specimen, and we'll talk about how to do that in a minute here, then you can take some more detailed photographs. And the 3 photographs that we're going to recommend that you take is one of that face and cheek area, a picture that shows us the top of the thorax so that we can determine, see that horseshoe shape. And then we want to see a clear photo that shows the 5th and 6th tergal segments of the bee. And to give you a sense of what the face shot would look like, this is Franklin's bumblebee. You can see that, like the Western bumblebee photo that I showed you earlier, this species has a short cheek, or is a small malar space. And you can also see, in addition to that, that this species has a lot of sort of shorter yellow hairs intermixed throughout its entire face. While from afar it may look black, there are actually yellow hairs that are intermixed throughout its face. On top of that, its vertex, or the top of the head, is yellow in this species with some minority of black hairs intermixed. But as much detail as you can get in a photo about the face will certainly help with identification here. And then here's a photo from the top that shows the top of the thorax. You can see that horseshoe shape where instead of just being at the front of the thorax, you can actually see that extends, sort of that yellow extends so that it's between the wings with that black sort of dot that you could imagine in the center cut out of it. And then the last photo that we would like you to take would be of the back of the abdomen here. So the tergal segments 5 and 6. So we can see those pale white hairs and that there's some black intermixed in this area, at least here. Those photos are going to help us identify this species. A very close lookalike for Franklin's bumblebee that overlaps 100% of its range, or at least did historically, is the western bumblebee. The species has a lot of similarities. It also has pale hairs at the back of the abdomen, although these pale hairs will extend further closer to the head. So these are also going to be on the 4th tergal segment. And from what I've seen in photos and specimens, this hair on Bombus occidentalis is a brighter white, or at least can be. This species is actually quite variable in its color patterns and can fade to sort of more of a cream color, which looks a lot more like I've seen in franklini. And you can see that in the color patterns that you see here. Sometimes it fades more to this cream color, especially this color pattern we especially associate with coastal California. So we really need to be careful with this in Franklin's bumblebee. But usually those pale hairs extend up into the 4th tergal segment. And if not, there's usually yellow hairs on the 3rd tergal segment. Again, for Bombus occidentalis, the key photos that we want to take are again very similar. We want to see a short cheek, so a picture of the head and face. And you can see that there's fewer yellow hairs mixed in on Bombus occidentalis' face, and the vertex in the species is also usually black. And then a photo that also shows the back of the abdomen, so that shows the pale or white hairs on the back of the abdomen is very important for this species. And then finally, similar to Bombus franklini, we'd like to see a photo of the top of the thorax. And remember, instead of a straight line here, Bombus franklini had that horseshoe shape. So the yellow hairs would have come all the way back to between the wings. Whereas in Bombus occidentalis, we really just find it sort of the shoulders of the bee, or that straight line that goes straight across, sort of cut off in front of the wings. And that's another distinguishing feature for occidentalis versus franklini. And just a little bit about the range. So this is the historic range and current range of Bombus franklini in Oregon, Washington, and Idaho. So the white are areas that we've conducted surveys over the last 3 years in those states, and the blue are where areas that we've detected the western bumblebee. So you can see that the western bumblebee is certainly overlapping in range with Franklin's bumblebee, and you could certainly expect to encounter it. So knowing how to distinguish between Franklin's bumblebee and the western bumblebee is very important. We've not done similar surveys in Northern California, but I would expect that the western bumblebee would also persist along that California-Oregon border on the southern side in California. These are the historic records of Franklin's bumblebee. You've seen these records before, but just want to show them to you again. This is the range where we would expect to find it. Obviously, we've talked a lot about this today. Since you're going to be out conducting surveys, we hope that you'll do comprehensive surveys that will look at the entirety of the taxa that are out there. And because of that, I want to bring your attention to this species. Leaf mentioned this species earlier when he was talking about cuckoo bumblebees. This is the suckly cuckoo bumblebee, or Bombus sucklii. This species is exceedingly rare. It's believed to use the western bumblebee as a host species, so it attacks nests of that species. But we're very concerned about it. We did not detect it at all in the Pacific Northwest Bumblebee Atlas from 2018 to 2020. We're still hoping to find it. And since you're going to be out surveying for bumblebees, we hope you will also keep an eye out for this species. Actually, the last known observation of this species was in Southern Oregon, not too far from Ashland, east of there. And I believe it was observed by Pepper Trail, who I believe is also on the call here today. So it's certainly possible that we could detect this species, and would be great to keep an eye out for it. So this is a cuckoo bumblebee, which means that Leaf showed us earlier that the hind leg of bumblebees has this corbicula in it for carrying pollen. Because the suckly cuckoo bumblebee is a cuckoo bumblebee, it actually does not collect pollen. It lets the host species do that. And so it's lost this corbicula from an evolutionary perspective, and now has this very dense, hairy hind leg. And that's how you can tell cuckoo bumblebees from true bumblebees, or at least one way that we can do that. And so if you know that it's a cuckoo bumblebee, we're also going to be looking for a black head and face. So the hair on the face below the antenna is going to be entirely black, as well as the vertex, the top of the head, is also going to be black. And so another photo that we'd like you to take for the species is straight on down, looking from the bee, the back of the bee, the last tergal segment. If you can take a photo on females of T7 here, the suckly cuckoo bumblebee has these what we call keels. They stick out to the side beyond. So this is actually the sternal segment, S7 or S6, excuse me, sticking out to the side of T6. And this is one of the diagnostic features for the suckly cuckoo bumblebee. So getting a straight down picture of that last tergal segment is something that would be very helpful for determining this species versus its lookalikes. Another thing would be just a straight on picture of the face and head to differentiate Bombus insularis from Bombus sucklii. So you can see here Bombus insularis, while its face appears somewhat black from afar, it has these tufts of yellow hairs around the antennae. So again, taking a picture of the head and face, and then also a picture of that sixth tergal segment from the top are going to be the differentiating features that we really need to see to identify this species. Okay, so that's really, those are the species I'm going to introduce you to today, with sort of the knowledge that there are many more species that you could encounter, especially in this part of the world. It's a particularly biodiverse region for bumblebees, as Leaf mentioned, and you will find, maybe as many as 23 or 24 different species down in this region. So it's a great place to go bumblebee watching, and adding the search for franklins is just another thing to add to it, which is fantastic. I'm now going to talk somewhat briefly about how we would like you to survey for this animal. So if you can imagine that you're at a place where you've adopted a grid cell, you've picked an area out that you want to go look for this species, we're going to talk about how to do that. And if you'd like to officially sort of contribute to the Pacific Northwest Bumblebee Atlas while you do this, even if you're in California, we ask that you're going to set up a Bumblebee Watch account, and I'll show you briefly how to do that. You'll adopt one of these grid cells and then go out and conduct surveys. And we'll talk about how to plan that, how to collect the bumblebees, and then I'll show you briefly how to submit your data online. So the first thing you would do is go to bumblebeewatch.org. And if you haven't already, we would ask that you sign up for the project. You're just going to click sign up here. It's like any website, if you've done this before, where you're just going to sign up for an account and give yourself a username. And then once you have your username, you'll log in and you'll see your profile page. And one of the cool things about your profile page is it actually shows you a list of all of the bumblebees that you've had verified on Bumblebee Watch, which is kind of a fun thing. You can keep your lifer list there. And it is important to remember your username because you're going to use that when you adopt your grid cell. So once you have your Bumblebee Watch account, then you can go over to the PNW Bumblebee Atlas .org/FFBV, and that's where you can go to adopt a grid cell. Use that map that I showed you how to use earlier and how to interact with it. And you can, right below this, if you scroll down, I showed you that form that you would fill out in order to sign up for a project and let others know that you intend to go out and conduct some surveys for the species. So that's sort of the base. And then once you've adopted a grid cell and you have a Bumblebee Watch account, you can go out and conduct your surveys. The survey window for this species and for all bumblebee surveys in this area is going to be June, July, and August. This is when workers of most species are active. We'd like to catch workers versus queens just because it's less likely to have an impact on the colony. And males, you can probably, it didn't explicitly say this when I was going through it, but I was talking about identification. I was mostly talking about female identification, which are much easier to identify from photos than males are. That doesn't mean that you shouldn't photograph males. It just tends to be more what we talk about. But please still photograph males. We'll do our best that we can to identify them. And it's easier in, say, Franklin's bumblebee and the Western bumblebee than it is with other species like Bombus vosnesenskii and Bombus caliginosus, whose males are almost indistinguishable from photographs. Optimal conditions will be basically when you're comfortable. 60 to 90 degrees Fahrenheit are the best temperatures. A little bit above that is okay. A little bit below 60 is probably also okay, particularly if it's sunny. But if it gets much above 90, above 95, I wouldn't survey. It's going to be too hot. Bumblebees are going to be in their nests trying to regulate the temperature of the nest more than they're going to be out foraging. And then ideally, we're going to survey in relatively light wind conditions and no rain. We've talked some about which areas are the best to survey. For this species, we really want to look everywhere. So natural areas, urban areas, public roadsides and rights of way. But again, make sure you have permission to survey where you are and follow all posted rules and regulations. We've talked that BLM and Forest Service land are really the best places to survey. State parks and state wildlife areas are also an option, although you will likely need a scientific collecting permit for these. I'm in the process, like we just talked about with CDFW, of getting a permit to survey in some Oregon state parks, but that's not in place yet. But if you're interested in surveying in some of these areas within the range, please let me know and we can work on it. So anyway, just generally make sure you have permission to work where you are working. The materials that you would need to conduct the survey are listed here. There's data sheets that are available from the Pacific Northwest Bumblebee Atlas website or in the volunteer guide that we have. And we can put the links to those in the chat. An insect net is very helpful in vials, a cooler with crushed ice, a camera and a smartphone or a smartphone. We've found that the pictures taken with smartphones can be somewhat spotty at best. They can work in a pinch, but if you have a dedicated camera, we do find they work better. And then some way to record your location, a timer to record time. And again, your smartphone can be used for a lot of this. And then some ability to grab a weather station nearby. So you can use a phone app, or you can actually grab weather data once you get home from a nearby weather station. And then field guides can be helpful, and paper maps are also a good idea, especially since some of these areas are going to be outside of cellular range. So be aware of that and planning ahead and making sure you've thought about those contingencies in terms of your travel plans and your ability to navigate outside of cell range. So after that, you're going to go through this workflow where you're going to conduct a survey. And again, this is an area that I'm not going to show you this video today, but we can share the link with you for this photo, or I will share the link of this photo of the appropriate video for you to do this now. Just pulling it up. Here it is. So we have a full video here of what it's like to conduct a survey. And this is for basically how to net bumblebees, how to chill them. It'll walk you through the entire process, how to put them in a cooler and how to photograph them. I'm going to talk about some of that in the few minutes that I have ahead of me here, but that video that I just shared there is really your best resource in order to, if you're new to this project and want to learn how to do a comprehensive survey, the video that I just shared will help you to do that. Let me get back to my presentation here. Okay. So the most platinum version, or the best thing that we believe that you can do to conduct a survey for this project, is to do what Jeff said the Fish and Wildlife Service are doing, is to do what Pacific Northwest Bumblebee Atlas volunteers are doing, and to do what the Xerces Society are doing when we're out conducting surveys. And these are, within your grid cell, are to pick and plan a 1 hectare size plot in which you're going to conduct a survey. A hectare plot is about 2.5 acres. It's about, if you think about the length of a football field, 120 yards long. It's about that length square. It's also about the size of a soccer field or an inside of a track, the infield of a track. All of those are about a hectare. And it doesn't need to be exact. You don't need to measure it out, but this is just about the region that we're conducting our surveys. And then you're going to conduct what's called a 45 person minute survey. And a person minute means that if you're by yourself, you're going to survey for 45 minutes. If you brought 2 people, you're going to survey for 22.5 minutes. If you brought 3 people, you're going to survey for 15 minutes. So basically, you're going to have 45 minutes of searching, but they're person minutes. So if you have 3 people, you're not going to end up with sort of 135 minutes of surveying. We want them to be equal effort so that they're comparable across different regions. And then you're going to capture every bumblebee that you encounter during those 45 minutes. And you're going to put that bumblebee into a jar. And then you're going to put that jar into a cooler with ice. And the purpose of putting the bumblebee into a jar and then onto ice is to initiate cold anesthesia. It essentially shuts the bee down and it makes it immobile so that we can take the detailed photographs that I've shared with you before. And I'm going to talk about safety measures with this in a couple of minutes, about the best and safest ways to be chilling bees. Since we're thinking about an endangered species, we want to be really careful as we do this. I put a star here because it says capture all bumblebees. If you're in an area, like I've been in some areas down in the Cascade Siskiyous where there's a ton of vetch or a ton of highly attractive plant, and there are literally thousands of bumblebees, that you can hear all around you. And if you caught every bumblebee that you saw in 45 minutes, it would be, you'd go crazy, and you'd get a lot of bees that would end up being the same species. So if you're in that scenario where there are a lot of bees that appear to be of the same species to you, we ask that you grab 10 lookalike species or 10 lookalike individuals, put them in jars, put them on ice, and then at that point you can switch to grabbing individuals that you think look different. And the reason we grab 10 is if there's likely to be, if there are lookalikes within a meadow, we found that around 10 of them would capture those lookalike species. So that's the protocol if you're in a highly abundant meadow with a ton of bumblebees that all look very similar to each other, is to grab 10 of them and then focus on capturing for diversity. This is our preferred survey type because it captures a picture of the entire bumblebee community. And we're also going to grab lookalikes that you might otherwise miss. And you might also grab other target species for this project. Morrison's bumblebee is on the fringe of this range. Suckley's cuckoo bumblebee is certainly within range, as is the western bumblebee. So please do these surveys if you can. And then the efforts will also be comparable with other Bumblebee Atlas efforts, including the California Bumblebee Atlas, once it launches either later this summer or next summer. Sort of the next thing I'm saying is what sort of a gold survey effort, if you can't do this, would be to go out and just conduct a timed survey for 45 minutes. Now that you know more or less what the species look like, you're going to go out and just look for this species, again, within a hectare, but you're only going to look for and capture bees that look like Bombus franklini. But again, best to use ice to initiate cold and anesthesia and to take the detailed photographs that we need. The pros of this is that it can happen sort of anywhere at any time. You can cover a lot of area with a lot of volunteers with an effort like this, and there's a relatively low time commitment. Oh, sorry, this is, I was supposed to be talking about a completely different survey effort. That was the gold, which is great. Now we're talking about the silver survey effort. So apologies for any confusion here. This is sort of where you're just going to be opportunistic. You might be on a hike, you might be in your backyard, and you might think you see the species and you're just going to snap a photo of it and you're going to share that photo with us. And the best, this is great because it can happen anywhere at any time, and you can have a lot of people participating. The bad parts about this is we don't get effort associated with it, and the surveys tend to be biased towards common animals that are fairly easy to photograph on flowers. So these are not our recommended, but if this is all that you can contribute, then that is fantastic. You're going to submit all of your data to bumblebeewatch.org, and I'm going to put a link to that into the chat. You're basically just going to go there. Once you have your account, you will go to submit your data, and at the top of the page there, if you've conducted one of these comprehensive surveys that's 45 minutes long, if you've done sort of the platinum version of this, you're going to select Pacific Northwest Bumblebee Atlas at the top of the page as your project. And then you're going to enter a whole host of data about the habitat in addition to the bees that you found. Your silver survey efforts and your gold survey efforts can also go here, but you would use the project code Bumblebee Watch there because you're just going to be sharing opportunistic observations of these animals and not the habitat information associated with them. The safe handling procedures for best practices for bumblebees, for handling bumblebees. If you are walking around in your hectare plot and you're capturing bumblebees and putting them into plastic or glass jars, it is very important that you do not leave those plastic jars outside of a cooler in the sun. You'll get a very quick greenhouse effect happening within that jar, and the bees can overheat very quickly. So please do not leave jars in the sun outside of a cooler for more than a couple of minutes. They should go, basically it should go from jar or from net to jar to cooler as quick as is practicable. Sometimes you might be walking back through your plot and encounter another bumblebee and capture it, and that's okay. But you just don't want to do that for 20 minutes or something like that. You want to sort of a maximum of 5 minutes of a bee being in a jar by itself is best practice. Once a bee is on ice, we believe that it's safe to keep those bees on ice for up to 2 hours. And I've had results that are fine, actually longer than this, as have many others. But 2 hours is a nice practical guideline to set for yourself. That means if sort of in a 45-minute survey, if Franklin's bumblebee, say, was the first bee that you caught in your survey and you put it in the cooler, and then you did your 45-minute survey, and then you started processing your bees and you had 25 bees to process, it might be 2 hours before you have taken photos of that bee, but this allows us enough time to have that entire practicality. That doesn't mean that you should keep it in there for 2 hours, but that would be the maximum amount that we'd want to keep a bee on ice. We recommend using crushed ice or iced cubes, not dry ice. And if you're at home, please don't use your freezer. Freezers and dry ice are just too cold, and they can actually they can be lethal and kill bees. So be careful and try to use something that's not that cold. Crushed ice is the best option. With properly chilled bees, you should have several minutes to take the photos that you need to. So you've got plenty of time under normal circumstances, but it's a great idea that before you take that bee out and start letting it warm up, that you get your camera ready and you get the settings set and that you've tried it out and things are gonna work really well. So take some time to set up your station and get ready before you start taking bees out of jars and out of the cooler. And then once you're done and you've taken all the photographs that you need, you want to release the bees pretty close to where they were captured. So within 100 meters. On hot days, especially, let bees warm up in the shade and not the direct sunlight. They'll warm up plenty fine on a warm day, and putting them in the sun might add additional stress. So just let them warm up in the shade, and it may take a few minutes, but eventually they will warm up and fly on their way as if nothing ever happened to them. So those are really the best practices. If I'm going to boil this down into 4 sort of bullet points, bees should ideally not be out in vials outside of a cooler for more than 2 to 3 minutes. They should be processed within 2 hours of capture, so not be on ice more than 2 hours. Please don't use dry ice or freezers. They're too cold. And release bees in the shade within 100 meters of your capture, or close to that at least. Don't drive them several down the roads to a picnic table you saw to take pictures of them. Please do it at the site where you conducted your survey. And then also along the lines with safe practices, and Hilary alluded to this, is to clean our materials between bees. So if you're using jars, Leaf mentioned, and others have mentioned that the leading hypothesis for the decline of the species is probably related to a disease, a fungal pathogen. And the last thing that we want to be doing is transferring disease between bees within this region. So between surveys, particularly if you're changing locations, it is best to sterilize and clean your jars and lids between survey locations. And this would mean using a 10% bleach solution, rinsing them and allowing them to dry, or potentially using alcohol or hydrogen peroxide spray, or potentially using, if you're in the backcountry or away for a while, you could use alcohol wipes, and just allowing these to air dry and get any sort of scents or other chemicals out of there, leaving enough time to allow them to dry and air out is really important. In a pinch, if you don't have these chemicals, you could use hot soapy water. But in this case, we would want to manually scrub them and not just let them soak. So you'd want to get a bottle brush or something similar and then rinse them and allow them to dry. So those are the best practices. Likewise, Hilary also mentioned this in her presentation. Please avoid harming or killing Franklin's bumblebee, and do whatever you can to avoid this. Photo vouchers are really the best and only way at this point to verify the species. No take is allowed in Oregon, no take is allowed in California. So please focus on photo vouchers. We can get enough detailed information to identify the species from photos. That said, accidents happen. You may kill a bumblebee. It happens. I think what I've heard from the Fish and Wildlife Service and from the California Department of Fish and Wildlife today is that this is not an ideal situation to end up with a dead Franklin's bumblebee, but we certainly, if you found one and killed one, we need to know that it was found. That is an important thing. And I don't think that that you'd be at risk for letting us know that this happened. This would be an accident. This would be, and the information that we would gather from that would be much better than not. And Jeff, it looks like Hilary's maybe jumped off, but Jeff, if I'm wrong about this, please chime in and tell me differently. But I believe that we're going to We want this information, and certainly we're not going to prosecute somebody because they accidentally killed a bumblebee while surveying for it. And certainly the information we would gather about location would be highly valued. So please, notify CDFW, the Fish and Wildlife Service, and the Xerces Society if you find this species and accidentally kill it. Obviously not an ideal situation, but these things can happen. And then again, just as we're photo documenting these species, I've talked about it, but we want a clear picture of the head and the face that shows the vertex as well as the hairs on the face, a profile that shows the thorax and between the wings and ideally below the wings as well, can be diagnostic for some species. And then a picture that shows all of the tergal segments on the abdomen. And we really want to make sure, sometimes when you get these profile shots, the bees will curl up and you can't see T5 and T6. So please be really careful, especially for franklini, to take a really clear photo of T5 and T6 so that we can see those pale hairs. We don't want them obscured by a curved abdomen. And then finally, please be really careful when you're taking photos. We get one chance to do this, right? If you go out and conduct a survey and you spend hours driving or hiking to a site and you capture bees and you put them in a cooler, and then you end up taking photos that don't allow identification of the individual, it would be a true shame. So take the extra couple minutes it'll take you to practice at home and make sure you know how your camera works and how you can get the settings and the focus working so that you can get photo in bright light conditions and dark conditions. Experiment and try and make sure that you know your camera. And likewise, if you need to get your hand in there to pull the wings apart gently, or a pencil to pry them apart to take a picture of the abdomen, that's okay. You can do that. They don't need to be perfectly composed photographs that are beautiful to go on a wall somewhere. We really just need to see these features. And so taking the extra time to making sure the lighting is correct and and the white balance is okay is really, really important. It's much more important that you be a good photographer for this project than it is for you to be a bumblebee taxonomist. You can leave the bumblebee taxonomy to Leaf and I, and we can handle that side of things. We just need you to be out there doing these surveys, generously volunteering your time and taking these great photographs and sharing them with us on Bumblebee Watch so that we can hopefully find Franklin's bumblebee. And that is all the material that we have for you today. Again, I apologize for going over and past 4 o'clock, but I feel like it was the right and best thing to do, as all that material was really important to share. At this point, I'm happy to take any questions. And as I take questions, what I'm going to do is I am going to launch the poll. And the poll here is going to be the quiz, which will serve as your ability to be listed on the Xerces Society's California Department of Fish and Wildlife Service permit. So this would allow you to conduct surveys within the state of California for this project. And I'm happy to launch this poll, and this is not supposed to serve as a barrier for your participation. If I launch this poll and you don't know one of the answers to the questions, you're welcome to drop it in the Q&A and we'll address it right now and you can answer it correctly. This is really just so that you know the protocols and you can show Xerces and CDFW that you've been trained and you've thought about this and that you know how to do this safely. And that's the only reason we don't want this to be a barrier. But the Xerces Society will be liable somewhat for your participation. So please, take the time to answer these questions and get your name on the permit. If you have any questions about this process, please email me. I dropped my email address in the chat earlier, or Hilary, who also shared her contact information. So I'm going to go ahead and launch this poll. If you're interested in being named on the CDFW permit, please go ahead and answer those. You don't have to if you don't intend to be named on that permit. And with that, I will see if there's any questions we need to address. And if anyone needs to run, I guess I'll just say thank you very much. Now, when you leave, I think you'll get a link to an evaluation form. If you take a few minutes to fill that out, it really does help us improve these programs as we move forward. So thanks very much.
Rich, there are two good questions from Pepper Trail in the chat.
Okay.
You might be interested to answer. One says, say there are lots of bumblebees, almost all Bombus vosnesensky, and you capture 10 and 15 minutes. Do you then spend the next 30 minutes just looking for other species? And if you find none, you call it good with your 10. That's the first question.
Yeah, that would be the practice that I would recommend. Yep. And, you know, yeah, I mean, I've seen those surveys, Pepper, so I know what you're talking about for sure. Yeah, that's what I would do. I think you're good at 10. You know, maybe if you've looked for 25 minutes and you get a chance to grab 5 more or something like that at the very end of your survey, that would be great, particularly down in that region where you've got Caliginosis, potentially Vosnesensky and Van Dykii, all which can look, as you know, as I know you know, can look very similar. So you know, maybe up it to 15 at the end of the survey if you haven't seen anything different. That's a good question.
Rich, several people are saying the poll has disappeared while they were filling it out. Do you want to relaunch that? 3 people are having trouble.
I'm a little nervous about this because I just clicked relaunch and it said, well, clear existing polling results, which means that anybody that has already participated in this may have lost that. It looks like that's only one person though, so I'm just
a single person, yeah.
I'm just gonna do it, and if it's you that I've kicked out, then I'm sorry.
Go for it.
Okay, thanks for letting me know that, everybody. Was there another question from Pepper Leaf?
Yeah, the question was basically if so Pepper has adopted a Pacific Northwest Bumblebee Atlas grid cell that is it's coincident with one of our Finding Franklin cells, and the question is, does it matter how I enter data if I do a full platinum survey? So if you're collecting for PNW already, yeah, go ahead.
Yeah, no, you don't need to readopt an FF grid cell, Pepper. If you already have an OR grid cell that overlaps with those, you can just continue working. You're certainly welcome to if you'd like to. I mean, we may send particular emails that are directed towards people that have just signed up for this project. And if you want to, you know, adopt it so that you make sure that you get that communication from us, then feel free to adopt a FF, the same FF grid cell. But yeah, you'll just submit your data as if you conducted a Pacific Northwest Bumblebee Atlas survey.
Here's a question from Adrian, who says, great webinar, but I have a ton of questions. I'm interested in a bit more clarification about quote unquote vertex, which is a term that's not defined in Williams at all. Is this only anterior of the ocelli, also behind them? This is a great detailed question, and it's good advice for me because we are slowly thinking about putting together a second edition of the book, and I'll make sure that we address that issue. The vertex is, you can think of it as the forehead of the bee. I cannot tell you off the top of my head if it's strictly anterior of the ocelli, which would mean in front of those small light sensing eyes on the top of the head, right? Versus also behind them. Generally,
I think it's almost entirely posterior to the ocelli.
Yeah, I think that's right. I think it's almost entirely behind them. But you will often see people refer to it in taxonomic stuff, and there's a little bit of a dodge, or they're extending the definition to really mean the top of the head, generally speaking. So Rich, here's another good question that I was going to answer in the beginning in the first module, and I didn't say it. Karen asks, is the only honeybee in North America the European honeybee? You noted that only one species makes honey, so there was no honey made in the wild before we brought in honeybees. I'd always thought wild honey was original to this continent. Karen, that's correct. Apis mellifera, the Western honeybee, is not native here, was brought here in the late 17th century, I believe. Actually earlier than that. There may have been early 17th century. Anyway, so there were no honeybees making honey here before that time. Two things though, there are some other social bees that do store nectar and turn it into honey, although not found in the US and Canada. So for example, there's a whole range of tropical stingless bees that make honeys in Central America and in Southern Mexico. And also you may be interested to know that as recently as about 15 million years ago, which isn't that recent on the timescale that I exist in, but nonetheless, there were species of the genus Apis found in North America. And we know this from fossil evidence. So at one point there was honey on this continent and then it went away for a while and now it's back. Rich, there's this question maybe you could answer. Anonymous attendee asks, how does CDFW prefer to have reports of bumblebees?
I honestly think they'd prefer that you submit your data to Bumblebee Watch and then we'll send them an extract of the data. But that would be assuming you're operating under Xerces scientific collecting permit. If you have your own scientific collecting permit or if you plan on applying for a scientific collecting permit, they have their own portal where you will submit your data. It's one of the requirements of applying for an SCP is that you submit your data to them. And I think there are multiple ways to do that. But for this project, submitting your data to Bumblebee Watch, and we will regularly share and update our information with CDFW.
Here's another one that's in the chat from Richard, and the question is, once we hit submit on the quiz, then what happens? How do we know the results?
Yeah, I'll have to go through those manually after this. And if you pass, so you don't have to answer all those questions correctly to quote unquote pass, but there are 3 key questions that we really want to make sure that you're answering correctly. And that was question 1, question 2, and question 6. We want to make sure you answered correctly. And as long as you have, you will have likely, there may be one more as well. I'm just sort of glancing through it as I look at it. But you will have passed and be approved, and I will send you an email letting you know that your name will be added to the scientific collecting permit. By answering that quiz, this quiz is not anonymous, so it's associated with the email address that you will have registered for this webinar with, and I will have that information, and I'll email you if you've been added to the scientific collecting permit or if you're eligible for that. And I would hope to be able to do that by the end of the day tomorrow. I see a question here. Is the permit only for Franklins? No, the permit is actually for all of California's sensitive bumblebee species, of which leaf 6. Are there 6? I can't remember. I believe there are 6. So it essentially allows you to conduct surveys for bumblebees throughout the state.
Yes, 6 of them.
Yeah. Well, anything else? It looks like we've potentially addressed everything, and hopefully looks like about 41 people have taken the quiz, which is fantastic. That means that 41 people want to survey in California for Franklin's bumblebee, which is amazing. And thank you all so much for dedicating your time today to this webinar. There's only one way to find the species, and this is by training a group of people to go out there with nets and cameras and doing this together. So we certainly couldn't do without you, and greatly appreciate your time. And Jeff, I hope you're still on here. Thank you so much for joining us today. What you contributed was incredibly valuable, and obviously our partnership continues to be incredibly valuable moving forward as we work on the conservation of this species and other bumblebees throughout North America. So thanks for being here today. And with that, I think we will, oh, sorry, Leaf, go ahead.
I was just going to say thank you to everyone who attended, and apologies for running long, but we're really glad you're with us, and looking forward to seeing your data.
Thanks, everybody. Have a great afternoon or evening, and we will see you out in the field. Oh, and if in case it, I don't know if Jeff, you mentioned this. Did you want to mention what's happening next week? And or should, maybe I shouldn't say anything. I don't know if you want to make that sort of public or not.
Sure. It's every year we Fish and Wildlife Service, along with some of our conservation partners in the Ashland, Medford area, host what we loosely refer to as the Bumblebee Blitz. And that's taking place next week, which is a whole week earlier than we traditionally do it. The peak of colony activity for Franklin's has always been, you know, if you look at that bell curve, around the third week of July. So ideally that's when peak detectability would be, because in theory you'd have more of them out on the landscape. And we moved it up this year by a whole week based on the ridiculous drought and incredible heat conditions that are out there, and after scouting some of the locations about a week and a half ago. And so next Tuesday morning, a big group of folks are meeting at Mount Ashland, which is that last known location for Franklin's Bumblebee, at 9 o'clock in the morning at the big rectangular parking area just past the ski area on Mount Ashland, basically at the end of the pavement before the first campground there and before the gravel road takes off. So we'll meet there at 9 o'clock. We'll do a quick round of introductions. We'll talk about some of the stuff we covered today, have a brief discussion about safety out in the field, and then head up to the Grouse Gap area where the Pacific Crest Trail crosses Forest Service Road 20, and spend all day, the rest of that day and all day Wednesday, surveying that area pretty intensively, looking for Franklin's and Westerns too, of course, and maybe even Suckleys if we're lucky. So it's a big group effort. It's open to everybody. There's usually a pretty big federal presence between Fish and Wildlife Service, Forest Service, BLM, but a wide variety of other conservation stakeholders participate in that. We've got local landowners, we've got other conservation entities like the Southwestern Oregon Pollinator Group, the Understory Initiative, a wide variety of other folks. So if anybody needs information about that, specific details, latest road report considering the construction on I-5 up there, or anything else, just shoot me an email. I'll be checking messages all through the weekend and on Monday morning before I leave. So thanks in advance. If anybody is coming along, it would be good to see you up there.
Great. Okay, I think we'll leave it at that. And again, thanks to everybody that participated, and thanks to you, Leaf and Jeff, and for Hilary and Laura as well for joining us. So have a great afternoon. Have a great weekend. Go Bumblebee watching. Have a good time.
Everyone.
Thanks, everyone. Bye.